Good morning and welcome to one of the probably most biology. Today I will talk to you about oxygen supersaturation, which is the oxygen enrichment in the water above the saturation level, a condition that recurrently and naturally occur in the field, especially in a highly productive habitat, and is generally neglected by experimental biology. And indeed, being breathing vertebrates, so we tend to assume the steadiness of a gaseous system as the target for a certain habitat. And this brings us to overlook the natural variation of the gas in the system and getting a sort of steady condition as the objective of our representation of the system. But this is a sort of an anthropogenic bias because tend to assume to describe oxygen in the field by means of averages. That can be daily, monthly, yearly averages, or using a scale larger than kilometers. So using average, this can bring us to depict the interesting trends, but which are sometimes not really meaningful for the communities in the system, because if we downscale to a much larger temporal resolution, we can find more interesting and more reliable variation of the system. I want to show you some real example that we collect in the field. These are comes from a meteor station in the Venetian lagoon, and the red line represent connect a monthly average of oxygen in the field, and as you can see, there is a quite evident variation during the 2 years time. But if we go further down to a larger temporal scale with an hour resolution, this is the pattern of the oxygen in the systems. And the picture becomes dramatic and more ecologically relevant for the community inside of the environment. You can clearly see variation of the occurrence of hypoxic condition and a seasonal variation. The larger range are representing warmer months where oxygen variate to a larger extent and to a smaller extent in winter months. And what is really, really interesting is the amount of time the system is supersaturated. So the amount of data are above the saturation threshold. If we zoom to one single week, this is a typical pattern of a productive system. The red line represents a daily average, but is almost meaningless for the community in the system, because they experience a much larger variation of oxygen, ranging from hypoxic condition to supersaturation condition. And this is a cyclical alternation of hypoxic and superoxic. This is what really shaped the life in such productive environment. I'm just talking about a temporal heterogeneity, but this is even more complex when we consider the spatial heterogeneity. And I want to mention that I'm working in the Venetian lagoon, not just because probably it's the most famous lagoon in the world, but also because it represents one of the reds in the Diaz and Rosenberg paper of the 2008. This is one of the hypoxic dead zone we studied so far, which is very famous. And our comprehension of such dead zone become more realistic if we get in the detail of the oxygen variation in the system. And these are the representation of the oxygen data from 5 different meteor stations in the system. As you can see, there is a strong variation in the oxygen Each of these bar plots represent about 10 years data, so they are quite consistent. And what is interesting is that roughly about 25–30% of this data stands above the 100% threshold. So there is about 1/4 of the time the system is supersaturated, and we can just subsample this supersaturation condition, and these are the distribution of the data of the supersaturation above the 100%. And what is interesting is the amplitude of this variation vary depending on the location. Is narrow in a more anthropic habitat like downtown Venice, but it increased dramatically in more natural and healthy environment. Mediterranean and Atlantic Ocean, and here also in the Indian Ocean. This is the Red Sea, and we sampled 3 different environments: the mangrove, seagrass, and coral reef. And the pattern is consistent. In such productive habitat, there is a strong variation of oxygen from hypoxic condition to superoxic, and a large amount of data stands above the 100% of saturation. So the water is always enriched by oxygen, alternating from hypoxic to superoxic. An interesting feature of the oxygen supersaturation is the periodicity. It has a period of 1 day. This wavelet analysis showed that there is a perfect significant period of 1 day. So every day the supersaturation condition occurrently again and again. And this is very important from a biological point of view. Another feature of supersaturation is the synchronicity. In this case, with the temperature, we collect the temperature and oxygen data here in the Venetian Lagoon and here in the North Sea in Scotland, at St. Abbs Marine Station. And just to close up to 3 days, this is the pattern of oxygen and temperature. They go perfectly in parallel, because the driver is the same. The light push the photosynthesis and rise the temperature of the water. So the two are perfectly in phase, and this is also underlined by this cross wavelet analysis. You can see there is a period of 1 day, and the 2 parameters are perfectly in phase. But most important is that this is a plot representing a coral reef data in the Red Sea. All temperature, whole high temperature during the day are matched by supersaturation condition. This means that the community in the system, when it is experiencing high temperature, is always experiencing supersaturation, never goes below the saturation threshold. And of course, we just wanted to briefly mention the driver, which can be physical driver, but also most important, the biological drivers. In this case, can be generally primary producers, so algae, phanerogams, microphytobenthos, and cyanobacteria. And this is quite interesting. You probably have seen on the coastline the bubble around algal mats or cyanobacteria mats, and these bubbles are pure oxygen released by the systems. So just to briefly summarize, I want to say that the oxygen availability, and most important, the supersaturation, is fluctuating with a dramatic day and night. They are predictable because the community can tune their physiology, can tune adaptive strategy on a predictable daily basis, and they are synchronous. Supersaturation is synchronous temperature. This is just want to mention another anthropocentric point of view. We will always refer, as experimental biology, we also refer to the 100% saturation in experimental setup. But 100% is just a condition that occurs twice a day during the fluctuation. It's nothing more than a broken clock that is right twice a day, meaningless for the community in the system. And of course, a a recommendation to reframe the experimental protocol adopting ecologically relevant parameters. I want to show you some experiment we did on supersaturation and the effect on select models. So we did in Red Sea some experiment on Talamita crenata, tuned crabs, very common there. And we saw, we retrieved that under supersaturation condition, there is a dramatic decrease of the critical PO2. That means that when the animals is exposed by the day to the naturally occurring supersaturation, it can withstand much better the hypoxic time, the hours during the night. So they can survive and endure hypoxic much better. And of course, and in parallel also can pay the oxygen depth. This is the lactate recovery, can pay the oxygen depth much better. So next day, after experiencing a large hypoxic exposure during the night, can pay the oxygen depth much more efficiently. We did also some experiment connecting temperature and oxygen, and to test how supersaturation can affect the thermal tolerance of the and we did on several species. Now this is just a coral reef fish, the Schilus, and this a typical mortality curve at the 100% saturation, so a full saturation. But if when it is measured at 2%, there is a clear increase of the thermal tolerance, and the curve shifts on the right side. And this is true for many different species. We tested 18 species of 4 different phyla, ranging from echinoderm, mollusk, crustacean, vertebrates, and in 3 different latitudes. So any system we tested at different latitude, different seas, is consistent. So when there is a high habitat, this creates a sort of fluctuating condition that increase the resistance and viability of the community therein. I want just to show you a couple of slides on the mechanistic explanation of this system. This is the oxygen consumption rate of Talamita crenata under full saturation. And of course, there is a clear increase of oxygen consumption. The metabolism rises exponentially and then drop when it becomes critical, the temperature becomes critical. But as Brad Siebel underlined yesterday, marine organisms can adjust their metabolism independence of the oxygen in the system. The metabolism can vary depending on the amount of oxygen they can withdraw from the system. And if we measure the same under oxygen supersaturation, a different picture emerges. So during the day, the animal is able to dramatically increase its metabolic rate. It can show a sort of temperature insensitive metabolism for a while, can increase the thermal tolerance, so can gain almost 2 degrees tolerance, and have such kind of extended metabolic performance during daily hyperoxia. Thank you very much. So I just wanted to conclude saying that it is very important to draft our experiment and our research field, including oxygen supersaturation in the equation. And of course, that the rate of a biological processes measured an average, adopting an average oxygenation does not really match the average of the same processes under realistic variable oxygen. And this is just a recommendation, since that the oxygen supersaturation and the fluctuation of oxygen in the field is driven by the primary producer, so by the vegetation, by the plants, algae, and other components. The conservation is a priority. Thank you very much.
Well, that was a great talk.
Thanks.
Denise?
Yeah, really interesting. I was just wondering if you've looked at things like suspension feeding bivalves and how that might affect filtration rates, and whether they can compensate or more than compensate for lost feeding opportunities by feeding at very high rates at supersaturation?
Thanks for the question. Yes, we did indeed some experiment on mollusk, and we did not test the filtration rate, but almost all kind of behavioral and metabolic pathway is accelerated or regained by under supersaturation, especially in the intertidal zone. This become really important to maintain a strong efficiency during the day. In that case, yes.
Okay, we have one last question. Do you have a? Sorry, yeah, one, and then we'll go to the last talk.
Hi. Thank you for a very nice talk. I could see, if you take back the slide where you have the sampling, water sampling compared to the measurements with sensors.
This one?
No, previous, you had the Winkler samples, no, further back. Yeah, or this one. Here is water, this is surface waters, right? So
no, it's about 2 meters in depth.
Okay, so yeah, but basically close to surface, and the water sampling was done by water sampling and Winkler titration, right?
No.
Okay.
These are made by optical sensors, so they do not need such a frequent calibration. They are the new generation probe made with the optical sensors. So
but the red dots, are they not from water samples?
The red dots are the average of the blue dots in certain sense.
Aha, so it's the average, okay.
It's the average. It's the monthly average, so I just collect all the data of the 1 days, 30 days, and this is the red dot.
Okay, because this is similar patterns we have from observatories running for many years, where the red dots basically represent samples from bottles. You take one sample every month or one sample every week. And consistently that misses out supersaturation, and that misses out low oxygen concentrations. And I'm wondering if we are doing modeling on oxygen, low oxygen zones, do we not need measurements because we never manage to measure and take samples at the right occasion. For meteorological forecasting, we have several hundred stations on land, and we do forecasting. We do measurements at 10 minutes. In the water, in the Baltic Sea, where I work, there is 2 or 3 stations where they measure, which are not consistent over time. And you try to do forecasting then with monthly data.
Yeah.
Is that enough?
The point is that there is no right time to take oxygen measurement. The only solution is to take at least a couple of days of continuous measurement and then understand the system.
Thank you very much. So our next speaker and last in this section will be Natalia Gallo, and she's from the University of Bergen.
Hello, yeah. To advance. Okay, thank you. Thank you so much. It's a real pleasure to get to be the last talk during a really wonderful session today. So today my presentation is on mesopelagic and demersal community responses to rapid deoxygenation and reoxygenation in a Western Norwegian fjord. I'd like to acknowledge my collaborators, Martina Sulos, Karl Bukowski, Lisa Levin, and Ana Grovea Salvanes, who introduced you to the fjords yesterday. But just a quick recap. So Norway has over 1,000 fjords along has its own unique attributes. It's really important to remember that these are still fjords, which means that these are true deep water ecosystems, but they have these really shallow sills that really prevent regular water circulation from occurring. The fjord we work in is called Moss Fjord. It's right here. It's about 50 kilometers north of Bergen, and this is also a sill fjord. And just appreciate the fact that the basin is about 489 meters deep, whereas the sill that the water has to enter over in order to replenish the basin is 70 meters deep. So water has to be of higher density in order to replenish that deep basin. And so warming and freshening along the Norwegian coast has shown to lead to less renewal frequency in the Moss Fjord basin water, which leads to oxygen loss and acidification. And if renewal doesn't happen for long enough, you can end up with anoxic conditions in the fjord. And fjords are also really important for salmon aquaculture, and salmon aquaculture is an additional contributor to deoxygenation. So if we look at Moss Fjord, the reason why Moss Fjord is such an area of interest is because it experienced this really rapid deoxygenation event. And it did so during a time where there were student cruises actually collecting ecological samples across this whole time series. So if we look back in 2011, Moss Fjord was well oxygenated and then experienced this rapid period of deoxygenation leading down to 2018. Notably, 2013 to 2016 was a period of really extensive aquaculture farming in this fjord. And then in 2018, the local government required that the aquaculture facility actually move because the conditions became hypoxic in the fjord. It's estimated that about 9% of that oxygen loss was due to excess oxygen consumption linked to salmon farming. And then just recently, this past year, there was actually a full reoxygenation event, and you can see that oxygen levels are back to 2011 levels. So this is the time series at 450 meters, so basically in that Moss Fjord basin water. Now, if we look actually through the water column and we look at how different components of the water column have changed through time, that's what you can see here. And basically, this is all relative to the oxygen concentrations in 2011. So here you can see the percent change in oxygen concentration relative to 2011. And again, the colors that you see here will be consistently used for all of the figures in the talk. So basically in the blue leading up to the red, this is the early period of deoxygenation in the time series. The redder points, those are the ones when the system was hypoxic in the basin water. And then in light purple, this is when oxygen starts creeping up and then is finally fully replenished. And so in the basin water, in these areas below 300 meters, we see a 60% decrease in basin water oxygen in less than 8 years. This is a really rapid loss of oxygen. In comparison, the Schmidt-Go et al. Study that we've heard a lot about in this meeting, right? Global oxygen levels have declined 2% over 50 years. And in areas like oxygen minimum zones that have experienced faster losses, you can see 4% losses per decade. Before I was working in the California current system, and there we saw a 30% decline over 30 years. That was a really big change. But again, in Moss Fjord was really substantial and really fast. And for that reason, it's a really good system to look at how communities respond to changes in oxygen. And because of the rapid pace of the change, we expect to see decreased potentially movement out of the fjord and fewer hypoxia tolerant species, or fewer hypoxia intolerant species. And so while we know that oxygen levels have changed in Moss Fjord, what we don't know is that if fjord communities actually are to these types of changes, both deoxygenation and other paired stressors. And so these are really the types of questions we're trying to get at with our research project. And we're able to do this because Anna has actually led a class for the last 10 years. It's a UIB class, Bio 325, where students go out to the fjords every September and they collect really nice depth stratified samples of this system. And so the data set that I will be showing you, they come from deep pelagic samples, which basically sample from 300 to 450 meters in the fjord. And then these additional depth stratified multi-net samples where the net has 3 cod ends and each cod end basically fishes a 100-meter depth interval of the water column. And so if we look at the samples that are available to us, for every single fixed depth interval, we have basically between 49 and 53 samples across this 10-year time series. And so what I'm going to do is basically take you through the water column, starting from the deepest area, moving up. And just some preliminary hypotheses our expectations here was because of how rapid that deoxygenation event was in an otherwise well-oxygenated system, we should be able to see clear community responses to this change. We also expect that those community responses will be most pronounced in the depths of the water column that experienced that most extreme loss of oxygen. So basically between 300 and 450 meters. And the expected community responses based on previous literature of effects of oxygen on community ecology are expected decreases in biodiversity, exclusion of hypoxia-intolerant species, and possible changes in total catch, as well as changes in community composition. So let's start at the bottom in this area that's experienced about a 60% decrease in oxygen across this time series. This is what we see first for total catch, for Shannon diversity, and for species richness. And I'll show all of the results kind of in the same format. So we see a very clear trend in an increase in total catch in the system. We see, along with that increase in total catch, we see a decrease in Shannon diversity. And remember that Shannon diversity captures both community evenness and species richness. And so here I'm also showing species richness to the right, which actually shows in recent years a little increase in species richness. So this really pronounced increase in total catch we weren't expecting, and this is something that we're looking into further. And additionally, the brief dip in species richness here may potentially be related to the strong deoxygenation. But there wasn't a really, really strong negative effect on species richness like we might have expected. Here I'm presenting a multi-dimensional plot of the communities. And so here every single point is one. And we do see some really pronounced grouping from year to year, but we see a much stronger driver of year as a change in the community composition of the system, with oxygen being significant, but a more minor driver of change. We also see certain species like merlangus, which are positively correlated with the oxygen axis. Basically, they show up when oxygen is high, and they're not there when oxygen is low. So now we move up a little bit into that next fixed depth interval between 300 and 200 meters. We see not a gradual increase in biomass, but just an increase in biomass in these most recent years following the hypoxic period in the fjord. Also, at the same time, we see that Shannon diversity decreases after this period of hypoxia. So basically we're seeing the opposite effect of maybe what we would expect here. And then we see species richness increasing slightly in recent years. Interestingly enough here, oxygen is not a significant covariate on community composition. But temperature is a strong driver, it appears, of differences in community composition in this depth zone. As we move up, so now we're in the intermediate fixed depth sample between 100 and 200 meters. Here, this is actually the depth zone that looks like the patterns most closely replicate the general patterns of oxygen in the system. But importantly enough, you actually look at what's happening in oxygen within this depth zone, it's not much. Likely any effects that we're seeing here may be being driven by animals that are deeper. Here we see that there's an inflection point basically in 2018 where we see our lowest total catch during that period in the basin water of the fjord. Similarly, for Shannon diversity, we see an inflection point with the highest Shannon diversity, and for species richness, similar to some of the other depths, see a gradual increase recently. So species richness appears to be increasing in the system in recent years. And when we look at the non-metric multidimensional scaling data, here is actually where we see oxygen having the strongest explanatory power on differences in community composition. So again, some of our kind of strongest oxygen linkages are in the part of the water column where oxygen did not change very much compared to the actual deep basin. We had a master's student, Carl Bukowski, who took a closer look at a specific component of the community. So here I've been showing you kind of the broader community picture, but Carl looked at 3 specific crustacean species that make up a really large component of the diel vertical migrators in the system. So he looked at Sergestes arcticus, Meganictiphines norvegica, our krill species, and Pasiphaea, which includes 2 to 3 species here in this group. But basically what he found was that Sergestes arcticus did show significant negative trends in abundance and biomass in the 2 deepest depth strata, basically showing that this species does appear to be sensitive to the declines in oxygen. But the krill species was less responsive than Sergestes, which may indicate that it's more tolerant. And what resulted because of that is that you actually ended up seeing a transition from a community that had more shrimp in it to a community that had more krill in it in this depth strata. Similarly, Pasiphaea, which is a more demersal and benthic associated organism, they showed a strong positive correlation with bottom dissolved oxygen conditions. So this is also an indicator that this is a species that is sensitive to the low oxygen conditions. So just to finish off with a few preliminary conclusions, we expected to see really clear community responses, and we did not. Sometimes we saw some interesting responses. Sometimes they were somewhat confirming of what we've seen from other systems, but overall we did really rapid decreases in biodiversity. And we did see some changes like this increase in biomass that we're still working to explain. We expected to see the most obvious oxygen-linked changes in that deepest depth zone that experienced the greatest oxygen changes, but we didn't find that. Basically, we see the strongest oxygen-related drivers in the shallower depth zone between 100 and 200 meters. And we did see some evidence of changes in biodiversity and changes in taxa, but they were, I have to say, more minor than I was expecting to see. So we're going to look further into what gives rise to the observed biomass increase. It's likely this jellyfish species, Paraphila, which has become much more dominant in the fjord in recent years. We're also going to look at contrasting fjord systems that are nearby that did not lose oxygen during the same time period as a contrast to see if there's kind of a system-wide effect happening or if there's something specific to Moss Fjord that was happening. And some really important things to consider are like the ecologically relevant time lag. So we had a really great keynote by Andrew Altieri yesterday, which basically shows that, you have to think differently about how a community responds to a stressful event versus how it recovers. And so here we have a great opportunity to look at how Moss Fjord recovers after this period now that it's experienced a renewal event. It's also possible that one of the reasons we see less of a response is that a lot of the community we're sampling, they're diel vertical migrators. And so their ability to move in and out of community level effects. And with that, I just wanted to let you know that we have 2 excellent posters from PhD students in our group that I encourage you to visit today to learn oxygenation in these fjords. And lastly, we are, for a global look at issues relating to climate change in fjords, we have put together a UN Ocean Decade satellite event that will be June 1st. And if you're interested, this is a QR code that'll take you to the website. And it should be a really great event. So thank you very much for your attention, and I'll take questions.
Thank you very much.
Or not.
We've got time for a question. Here you go.
Thanks. That was really interesting. I can't wait to get out there. My question is just how certain are folks that removing the aquaculture is what removed the problem? Because this has all kinds of implications.
Yeah, thanks for that question. So basically, Doug Oxness, who hopefully you'll meet during a visit, he took a look at basically the biological oxygen consumption in the fjord. And basically the biological oxygen consumption has increased during the period of really kind of extensive aquaculture in that fjord. We don't know that the slight increase in oxygen after they moved the aquaculture pen out was specifically due to that. But it certainly lines up quite well, and we don't have any other explanation for why oxygen increased in the system after they moved that system out. But the representative of that farm will actually be participating in the UN Ocean Decade event and giving us his perspectives on what it was like to be moved out of a fjord, low oxygen conditions. Yeah.
Okay, let's thank Natalia and all of our speakers. Thank you very much. It was a great session.
So we move now to the next session. So I'm calling the chair of the session. So we have Christine Da Silva, who is professor here at the Liège University. And we have online Moriaki Akasura, and Dimitri Teres.
Yeah.
Moriaki?
Yeah. Thank you.
Okay. Nice to see you online.
Good to see you.
Good to see you here. Yeah, so I will give the floor to them, to both of them, and Natalie, she's not there. Okay, but maybe she will come in the afternoon. So we have 4 chairs for this session. Okay, so 1 here and 1 at distance, and we have our keynotes, and I will leave Christine to introduce the keynote first. Thanks.
Hi all, thank you. So this morning we start a session on how the past can inform the future. And we will start with the keynote, Babek Kook-Dacker. Sorry for pronouncing.
Okay, thanks.
She's a professor of paleoceanography and climate change at the Lyell Center at GO2NE University, and she's interested in the relationship between global biogeochemical cycles and the evolution of the ocean and climate. And I guess she will give us some piece of that. So thank you very much.
Thank you, Anna. First of all, before I start, I wanted to thank the organizers for putting together a really nice conference and meeting. I've been really enjoying all the talks that have been so far. So the title of my talk is Fate of Ocean Oxygenation in a Warming World. This is a UK research and innovation funded project that is running at the Heriot-Watt at the moment. And I want to acknowledge all my colleagues working on this project with me. Okay. So first of all, why do we even care about what happened to oxygen concentrations in the past, right? What's happening is now is really important. And I'm going to try and touch upon why for me it's important. Then I'm going to show you some research from different kind of groups that's been going on. And this is followed by a discussion about proxy. So these are kind of the cornerstone, what puts all these reconstructions together. And finally, I will give you some what they're doing in the Fate of Ocean Oxygenation in a Warming World project, which we have abbreviated to Fargo after a really nice movie. So why, do we want to know how oxygen has varied in the ocean in the past? Well, one reason for me is because I think it can help us improve predictions of how oxygen in the ocean might change in the future. But there's also another reason, and that's from a kind of earth science perspective. So oxygen in the ocean is very closely linked to the organic carbon cycling with respiration and photosynthesis. And so any changes of oxygenation in the ocean atmospheric CO2, so you have a carbon cycle feedback there. But now to just go back to the first reason. So here we get the wonderful Schmidtko figure et al. Showing how oxygen has been changing since 1960. But I'm also showing another figure from Lothar Strammer et al. From 2010, which indicates how oxygen minimum zones have been expanding over the last 40 years. Now, as you know, models have been predicting that this decrease will continue well into the future, with the worst kind of IPC or greenhouse gas scenario indicating just change in the ocean deoxygenation. But what is quite interesting here is that the models actually haven't been able to capture the 2% oxygen loss that has been measured by Schmidt et al. And so the models are missing something. Now, Oshvis et al. Have suggested several reasons for this mismatch. One could be unresolved transport processes, variations in respiratory oxygen demand, or even missing biogeochemical mechanisms. And in the same paper, they kind of highlight, that we need dedicated observational programs. So I kind of take from this that we need to better understand how ocean dissolved oxygen and processes that influence it have varied on longer timescales. Now, obviously, we can do that by continuing observations, but this is going to be a very slow process. But another way to reconstruct dissolved oxygen levels and drivers. So what kind of research are people doing? The thing that we kind of need to realize is that in geological terms, the ocean's only been recently well oxygenated. It's only been about 500 million years. The whole water column has been fully oxygenated. Before that, especially deep was anoxic. So people over the last 500 million years have been looking at ocean anoxic events. So these are events where we've got what is evidence in the geological record of like organic layers that are evidence that, where these from that bottom waters were completely anoxic. And what is interesting is that these really big coincide with thermal maxima in the ocean, but also with major extinction events in the ocean. So now we're kind of moving to a completely different time scale. I would kind of look at glacial interglacial scales. So we've been looking at how oxygen concentrations have varied over glacial and interglacial timescales. And this is mainly in a way to kind of also try to understand how the biological pump has changed over those timescales. So this here is a figure from Sam Jack and Eric from 2012. And it shows you the difference between in ocean oxygen between the glacial, so that's about 20,000 years ago, and the early Holocene. And from this analysis, they concluded over this time period, there was an increase in deep water oxygenation. So during glacial times, more were poorer oxygenated than they are today. And on the other hand, in intermediate waters, they saw a decrease. So that suggests that during glacial times, these intermediate waters were actually better ventilated than they are today. Perhaps there weren't even oxygen minimum zones. Now, some recent group have done, and later on, people in Princeton as well, use different proxy methods or methods to kind of look at this. And at least for the equatorial Pacific, so around here, our research suggests that there weren't any changes in intermediate water oxygen concentrations. So that's, how different methods sometimes come up with different things. And one idea is that some of the methods from the previous analysis are a bit out of date. But obviously what we need to do is look at more locations in the Pacific and probably also in the Arabian Sea to kind of see what the global picture of this is. But what's most important is, for you, I think, is like, how on earth do we even come up with all these ideas of what's happening in the ocean in the past? And our source of information actually comes from deep sea sediments. So we have research vessels that go out and take huge sediment cores that they retrieve back. So these sediments have been accumulating over thousands to millions of years. And then we then take that back into the lab and take samples from it and do all sorts of things with it. So what kind of information is stored in these sediments? We've got geology, so that gives you an idea of environmental deposition event. There's chemistry that we can present in samples. And we can use all of this information as proxies for describing environmental characteristics in the past. A word of caution. So obviously your reconstructions are only as good as your location, where you get them from. So we want to target how oxygen concentrations have varied in the ocean. We need to kind of get cores from that location, basically. So if you start here or here, you might not actually get that. We don't know how dynamic oxygen minimum zones are either. So it could be that at another time, oxygen minimum zone was shallower, or what it was, sorry, shallower was up here, obviously, or deeper. So you kind of need quite a few cores along a transect to kind of get some information about that. And that's quite hard sometimes, because there's also locations in the ocean where you have strong currents and you have non-deposition. Zainab has done some wonderful work for that along the Peruvian margin. So 2015, Moffat et al. Came up with a really nice review. They gave an overview of all the proxy methods that are available to reconstruct oxygen concentrations in the ocean. And you can split these in traditional proxies, and you can split these in, look at bottom water and sediment oxygen and subsurface water. So we have laminations, and these are great because generally you don't find lamination when there's no biotubation occurring, because oxygen levels are so low that it just doesn't support those organisms. You also need seasonality in the kind of deposition environment to get these nice kind of layers. Another way is to look at benthic foraminifera assemblages. So these are tiny unicellular organisms in sediments and in sediments as well. And there, the kind of community composition often can tell you a little bit about, you know, metals that react differently to oxygen concentrations. And then for subsurface waters, people have been looking at nitrogen isotopes. And now, if you look at the whole kind of range of oxygen concentrations that we have in the ocean, it only really captures the lower range of oxygen levels. So we need to kind of do a bit better to kind of to cover the wider range, basically. So the aim of the Fargo project is to improve our understanding of cleaning. And our first package basically looks at method and using a multi-proxy approach. So we use the traditional proxies that I've just discussed, but we're also developing new proxies and really trying to do a multi-proxy approach to really see that we can get support from multiple proxies about what's happening in the past. So just a precaution, our work involves a lot of foraminifera. I've got some 3D printed models with me, which I will put out in a bit. But I just kind of thought I'd give you a little bit of overview of foraminifera because you don't know find out. So what are foraminifera? So they are single-celled marine organisms. They've got pseudopods. They can either be completely organic or they can have a shell, which is agglutinated or secreted. And they're mainly cell size, so tiny. Probably if you go to the beach and you get a sand sample, maybe not if you go to the Canaries because it's all volcanic, but in other areas you might actually have some nice forams in there as well. If you go into tidal areas, you'll find lots of forams, and the group from Angers know. But I don't know if you know this, but the building stones of the Egyptian pyramids contain giant foraminifera called nummulites, basically. So they are everywhere. So there's a specific groups of foraminifera that create carbonate shells. So they kind of precipitate these in the environment that they live in. And so within of environmental information. Now, but it's really important is that there's actually two groups of foraminifera. There's benthic foraminifera, so they live on the seafloor or within sediments, and you can find them almost everywhere around here. So generally you find more benthic foraminifera in shelf sediments than when you go to the deep sea, and I think that's just got to do with food availability and things like that. But planktic foraminifera actually float in the surface waters. And planktic foraminifera have a lot of promise to, to find out what's happening with oxygen minimum zones in the open ocean environment. Like I said before, if you want to, look at deposition in oxygen in sediments, you generally need to get cores that are located in these kind of oxygen minimum zones. But because planktic foraminifera live in the open ocean, they have got the potential to record variations in this as well. Although there's been that much work done yet. There's a talk, hopefully it's going to show a little bit of so what are we working on at Heriot-Watt? So we're working on proxies for bottom water reconstructions. One is the benthic foraminifera carbon isotope proxy. So this is a quantitative bottom water oxygen proxy that will range of 50 to 200 micromoles per kilogram. And we have Tommaso Paoloni and Sebastian Garrido working on this in the Lyle. And another thing that we're further exploring is something that was previously proposed by Anthony Byrne et al. And that's looking at pores in benthic foraminifera, especially on the umbilical side. And so this is species that live on sediment, the side kind of facing the water column. But we're also looking at surface water oxygen reconstruction. So worked with GO2NE Liu. He gave a keynote at the Kiel Ocean Deoxygenation Conference. And calcium and planktic foraminifera can basically be used as a qualitative oxygen proxy, and like a promise of kind of widening our research to the open ocean environment to really kind of see how, how oxygen minimum zones further on have developed. So now my next slide goes a little bit back to the panel session yesterday. So the Pacific really is one of the most understudied, undersoiled basins, I think, not only in terms of modern times, but this is the basin, and I think various papers allude on that, where deoxygenation will have the most dramatic effects on livelihoods, fisheries, etc. And just as an example, so this is a paper that was published last year by Mackay et al., and it shows you the first kind of carbon isotope transacts in the Pacific since the Kropnik kind of carbon isotope thing paper came out in 1985. So this is for the Southeast Pacific, so that's along here. And so you did a tremendous job getting all these new data together and using the carbon isotopes as a kind of bottom water, not a bottom water, seawater sea, basically. So we have like high carbon isotopes for water masses that are well oxygenated, and lower carbon isotopes for water masses that are oxygenated and high in nutrients. But what's really interesting here as well is that we have open ocean and then, and this hasn't been done for the Atlantic. What Dharma has shown is that you can actually, there's differences in the carbonized water in these areas. So mostly when we do reconstructing, when we use benthic formulae for carbon isotopes to reconstruct, comparing this with these open ocean transects, and this really shows that we need to kind of have closer to data for that. So that was just an aside. So my key message here is that there's many more bottom water and surface water oxygen proxies that are and have been developed. Only just touched upon what we are working on, but there's Julia Gottberg in Kiel who's working on uranium calcium ratios. There is people in Princeton looking at nitrogen bound to organic material and planktonic foraminifera. There's Jeroen Groeneveld and other people working on manganese, calcium foraminifera. So a lot is being developed here. And so the response, we've decided that we would have a workshop, kind of put all these developments of the new proxies together. So this will be in Bergen, just after the international conference in paleoceanography. This have very generously given us some money to support early career researchers to travel there. And so the workshop has actually got two aims. One is to write a review paper, and the other one is to kind of discuss PAGE's working group application. So this is a working group looking at oxygen concentrations have varied in different times in the past, basically. So if you're interested in PAGE's group working group application, just write something on the Padlet, and I'll add you to a list of people that want to do that. Now, planktic foraminifera have been extensively studied, but it's been mainly the planktic foraminifera that live in the sunlit ocean, so that's top 100 meters, that are a valid culture, and we've done loads and loads of calibration studies on those. Also, and Kate Davis has shown that quite recently, there's also foraminifera actually living in some areas with oxygen minimum zones, and we don't know anything about their biology or ecology. So we've recently written a SCORE working group proposal called FIERCE, so that stands for Foraminifera in Extreme and Rapidly Changing Environments. So that includes the kind of sea ice interface in the Arctic. And the idea is to kind of a sort of a scoping paper, knowledge gaps, and also to move forward and write proposals to kind of address this gap. So we're kind of hoping to have a little bit of like a forum traces network at some, but that's very ambitious, obviously. So yes, just to go back to the Fargo project, our aim is to improve understanding of the ocean oxygen cycle. I've already mentioned that we are developing methods for this, and we're applying a multi-proxy approach. So our second working package on hard at the moment is to create seawater oxygen concentrations, content, and drivers in the past. So for this, we've chosen 3 really important time periods. One is relation to the closure of the American Seaway. So the development of the Pacific oxygen minimum zone has been strongly related to the closure of the seaway corridor zones of the Pacific, kind of really well developed. And so we're trying to figure out exactly at what time oxygen concentrations actually start to feature in the Pacific Ocean. Now, the next time period that we're looking at is the mid-Pliocene warm period. And this is a really important period because temperatures on Earth were kind of as they are today, give or take a little bit. And obviously, but the oceans were equilibrated with the CO2 concentrations at the time. Now, the mid-Pliocene is important because it's a period when continental configurations were very similar as they are today. Basin topography was the same, mountain ranges were the same. And one important feature as well, and this is something that's predicted for further in the future, is that the Arctic was ice-free during summer. So this is something that we're working. Then the third time interval we're looking at recent interglacials. So basically, our current interglacial, but there have been many more interglacials in the past with different solar forcings and also with different greenhouse forcings. And to kind of find out what the underlying variability is there, it's really important to look at these interglacials as well. Now, in our third work package, we're actually integrating the information that we got through the reconstruction thing. So Vyacheslav Kohn is going to give you a little bit of a taster in the afternoon about the modeling that he's been doing with the closure of the Panama Seaway. HM 3 for that. And this is in collaboration with Birgit Schneider from University of Kiel and Alan Haywood in Leeds. So what do we hope to learn from this project? One is to get an idea of the sensitivity of Pacific Ocean oxygenation to warming. Another one is to look at what are the drivers of change and see what oxygenation in a warmer world. And then the third objective is to improve predictions of future ocean oxygenation. So there's a last bit. We're also trying to, we also really want to have impact and engage with politicians. So we've already set up lots of public outreach and activities. There's going to be a Future in Careers article in a couple of months, hopefully. Dharma has been, really champion citizen science in Chile. She's been going out there with Chilean nationals, river waters and coastal waters and explaining to them she's doing these things as well to calibrate proxies and things like that and to understand how ocean deoxygenation is work. We're also, through the SCORE proposal, we're doing capacity building with Chilean researchers. We're collaborating with the UPWELL program, which is about the Humboldt and how that's varied in the past, and the Chilean CLEP. And this is for the future. We're also hoping to kind of involve or engage with policymakers to kind of so that we can bridge research and policy. And then that lets me go to my last slide, where I say thank you to everybody for listening, the Fargo team and funders. And I will just put out some 3D models for Foraminifera. You don't have to come and see them, but if you want to, please do. And that's the end of my talk. Thank you very much.
Thank you very much for this very pedagogic and clear talk. Are there any questions?
Yeah. Oh.
Thanks for that. Very good. Your talk was titled "In a Warming World."
Yeah.
I wonder what do you think is the timescale that you can identify or can well really provide information from your sediments? Since in modeling studies we find that warming on timescale oxygen signals then warm after several hundred or a few thousand years.
Yeah.
And could you, or do you have proxies that could resolve these transitions, reorganizations of the ocean circulation?
There might be proxies, but we have the sediments that kind of have those timescales, right? Because generally, if you just get one sediment sample, that already represents 1,000 years. Yeah, it's quite hard.
What's the current feeling? What's the resolution you usually
So for the last 800,000 years, you can go up to maybe 5 to 7 centimeters accumulation rates per 1,000 years. But in the Pliocene, it's just up to 3 centimeters. So you get bioturbation and things like that. But I mean, I think it's also just a case of finding these really high accumulation areas, because recently in Newfoundland, they found, but that's the Atlantic. Our focus is in the Pacific, because this is where you have these big oxygen changes. In the Atlantic. There's such high oxygen concentration. It's very difficult to see if there's any variations in that level. Thank you.
I was intrigued by your delta C13 proxy. Can you say a few words about that?
Maybe you could talk to me afterwards, because that's very technical.
Thank you. Could you comment differences between the earlier results with the earlier, the proxies in 2012, where we had that OM, like subsurface versus deep result, and the newer results that are contradicting that?
Yeah.
It's because of the proxies, it's because of the location of the cores.
Yeah, just go back to
what should we, as modelers,
well, I think
aim for?
So unfortunately, there's not that many records with these new proxies. Yes, so the reconstructions that you see here, so you've got different symbols, and you've got, some of them are redox metals, some of them are from assemblages, some of them are trace metals, and a lot of them are bulk nitrogen isotopes. And the work by Struder et al. Basically is looking at planktic foraminifera-bound nitrogen isotopes, and that shows something very different. So they are suggesting that something happening in the sediments that affects the bulk sediments, but perhaps not the planktonic foraminifera. Have a look at their paper. That's kind of quite nice. The other thing is, if you look, there isn't actually that many spots as well either. And there are some, that are actually suggesting it might be a bit better in here as well. But a lot of those things are basically just nitrogen isotope records, bulk ones, which maybe are not as easy. So in general, I've been chatting to some colleagues in Edinburgh about that, and they say in general, for the nitrogen isotope records to work, you really have to be in the coastal area where there's high productivity and things like that. When you move over a bit further away, it's maybe not as good an indicator for it. So.
Thank you very much. I think we should go to the next talk.
Now we will start with oral presentations related with the session, How the Past Can Inform the Future. And the first speaker is Moriaki Yasuhara. He is from the University of Hong Kong. And he will talk us about shallow marine ecosystem collapse and recovery during the Paleocene-Eocene thermal maximum. So Moriaki, welcome.
Sure, thank you. Thank you, Dimitri. And welcome back everybody from coffee break. And today I'm talking about some example of time machine biology. We recently used this term, time machine biology, for paleobiological study, especially using sediment cores and microfossils to understand past, especially marine ecosystems and biodiversity. So Babette, great keynote, already explained about sediment cores. So I don't need to go into too much continuous historical record of biosphere or biodiversity. And we use microfossils inside of the sediment. Babette used foraminiferans, and we have different types of microfossils. And I'm ostracote specialist. So today's presentation, I use benthic ostracote as a proxy. There are abundant benthic microfossils in sediment core with high abundance, high biodiversity. And it's probably the only metazoan or arthropod or ichthyosaur or crustacean with enough abundant fossil record in small amount of sediment. So you can imagine beautiful marine ecosystems, different beautiful marine animals here. But among them, if we look into fossils in sediment core, we will find with sufficient abundance only microfossils like ostracods. So we need to use them if we like to understand past biodiversity dynamics in good resolution. So microfossils in sediment cores is a kind of model system or window to look into the past ecosystem. So we usually take this kind of approach as the time machine biology. And today's topic I'm talking about is PTM. It's a Paleocene, Eocene thermal maximum when our planet was much warmer climate than now. And this beautiful illustration is for high Arctic at that time. So it looks like pretty warm. And regarding future or ongoing warming and the oxygen decline, one major thing we are worrying about is habitat compression in shallow marine system. We saw an excellent talk this morning. It's actually happening. So oxygen minimum zone is expanding. So not only to the deep, but also the shallow depths. So shallow water habitat getting narrower with warming. Then organisms living there have narrower habitat, so could be at risk, for example, for extinction. In this talk, I'm showing this was actually already happened in the deep time past when temperature was much higher than present. And this is led to my PhD student, Sky, in collaboration with Mei Huang in the Smithsonian Institute and Fabien in Montpellier in France, and Marcy Robinson in US Geological Survey. And the location is US Atlantic coastal plain, this sediment core. And this is the result. You see the left-hand side curve, a kind of general climatic curve for PTM. So basically on the onset of PTM, we have rapid warming, and then we have recovery into the normal temperature later. And right-hand panels are mechanistic process-based modeling for origination and extinction and biodiversity. So this is origination and this is extinction. And this is biodiversity. You may ignore beginning and end. It's an analytical artifact. So we have big extinction at the onset of PTM. And after that, we have origination or recovery in the recovery phase of PTM. So we see the same signal. It's onset of PTM. We have diversity decline, and it's gradually going up again after PTM. This is different plot, but saying quite similar thing. Red species are extinct species. So you can think horizontal axis depth as age. So deeper depths mean older age, and shallower depths mean younger age. So it's core, depth in core depths. So red species gone at the beginning of PTM, and blue species as well for a while. But after the PTM, they came back. We call such taxa as Lazarus taxa. And some species came after the PTM. It may be came from somewhere, or it can be new species evolved. And this is NMDS plot showing faunal compositional change. So red is before PTM, and we have big faunal change get into PTM. And then after the PTM, we have gradual faunal recovery into original issue state, but recovery is not complete. So it's substantial faunal change and it didn't recover completely. And this is faunal change in different way. So vertical axis is depth or age. So deeper meaning older and the top is younger. And here is PTM and before and after. So we have shallow marine species before PTM, but they basically gone and replaced by deeper water species. And after PTM, there are some opportunistic species came in, and also some species came back, and also some new species appeared. It's a bit complicated. So this cartoon may be easier to explain the story. So this is before PTM, and this is during PTM, and this is after PTM recovery phase. So in PTM, we have oxygen minimum zone expansion. So deeper water species with oxygen sensitivity kicked out from deeper depths, get into the core locality. And then shallow species further kicked up to the shallower depths. And some warm adapted species survived there, but some sensitive species went extinct. And after that, oxygen minimum zone get into the normal extension, and then each of them can go back to original place gradually with some newcomers. So important thing is here, we actually had habitat compression and extinction. So oxygen minimum zone expansion caused habitat compression in shallow habitat, and some warm sensitive species actually went extinct. So this is exactly what about our future ocean. It already happened in deep time past. So it's important to study deep time data, paleobiological data, not only to understand deep past itself, but also to understand the present and future. And more broadly speaking, paleobiological data is quite useful to understand present and future. And this is conceptual cartoon based on different types of paleobiological data in combination with biological observations, showing past, present, and future marine biodiversity in tropical, temperate, and polar latitude. And red-colored hot water fish or animals going up to the north into the mid-latitude because tropical temperature is going to be too high. And orange intermediate temperature species going further up to the pole or Arctic to track their temperature niche. And green cold water species have no place to escape, so they probably go extinct if we continue this kind. And one important thing is tropical future, because tropical temperature is already too hot and even hotter in the future. Even for hot water species, they are escaping into higher latitude. So there are no replacement or no immigration into tropical ecosystem. So we will see much smaller level of biodiversity here in the future tropical ocean. Actually, yesterday's great keynote by Hans Portner and William Chen clearly show we already observe such thing in biological data. So in combining paleobiological data and biological observation, this is probably the future picture we most likely see if we didn't reduce any carbon dioxide emission reduction. Yeah, thank you for your attention.
We have time for questions.
Thanks very much. My question about the peak PETM is, can you rule out from the data or the information that transient phase during the reorganization of the ocean circulation, which means just a few hundred years? Is it possible to say, no, it was much longer, it was several thousand years? I think that would be extremely helpful for the modelers to know what to look for and what to calibrate the models against. Thanks.
Yeah, it's quite good point, and it's difficult question. And because my data is from shallow marine sediment core, so temporal resolution or time resolution is not too good, and the sedimentation could change a lot. So it's a reason why I show the data with core depth instead of age. So from our data itself, it's difficult to say time interval of actual PTM. And also, even in deep sea, PTM is a time of ocean acidification. So there are a lot of calcium carbonate dissolution. So age control is not too good. So I think it's quite difficult question. Maybe professional paleoceanographer there may know better answer, but it's my understanding.
Thank you. Maybe we have time for one more question, short one. Okay.
You talked about your, we were thinking about sort of sediment or north or slope to depth transects with Babette's talk, and about how that can sort of show you the expansion or contraction of an OMZ if you don't necessarily know where it is. And so I'm curious if you had any thoughts about maybe looking at sediment cores that were at higher or lower bathymetries than the core that you were looking at to figure out the real extent of that oxygen minimum zone and making sure it wasn't a feature of sort of some other oceanographic change that was causing the species range shift.
Yeah, thank you. It's a great point. Actually, I completely agree with you. And it's rather our interpretation based on available data from one site. And yeah, if possible, it's great. But in the field of paleoclimatology, people tend to get one sediment core in the best location rather than getting core in transect. So future, if possible, I like to do more in this area of US Atlantic coastal plain because there are several long core having PETM section. At this moment, I'm not 100% sure it's a depth transect or no, but probably I can have 2 or 3 sites there. So yeah, thank you for your suggestion.
Okay, thank you, Moriaki. I think it's time to move on.
Sure. Thank you, Dimitri.
Yeah, so today I will show you results of 2 of my studies focusing on the applicability of molybdenum and uranium as proxies for deoxygenation in coastal deposition environments. First, I would like to thank all the people who are involved in these projects. My talk is organized around 4 key questions. So starting with why should we care about using molybdenum and uranium as redox proxies? Then what can these proxies tell us? What are the limitations? And how can we explain and justify these limitations? So, well, we're all here together to study deoxygenation, so I guess I don't have to go into so much detail about this here. But we know that bottom water redox conditions are, bottom water oxygen concentrations are declining in vast areas of the coastal waters in Europe. And as we can see from a lot of blue area here, so this is where we don't have nice time series of monitoring data, is that where data is really lacking. And that's a particular case for areas that are more or less unrestricted
and also not suffering so much from hypoxia. And that's of course a problem because that we don't really know what's actually going on and how these areas could evolve in the future with climate change and eutrophication. So this is where sedimentary archives can help us to fill these knowledge gaps. And as we nicely heard already from Babette, molybdenum and uranium are really good tracers for that because they enrich in sediments. And as we can see from the study to the right, that throughout the Holocene, where we had several hypoxic intervals in the central Baltic Sea, that these hypoxic intervals correlate with increasing molybdenum and uranium concentrations in the sediments. So based on these findings, we studied the extent to which molybdenum and uranium can accurately reconstruct at 18 sites of 18 coastal deposition environments with different bottom water oxygen concentrations. And the focus here was on these mildly or non-reducing sites, which are vastly understudied with respect to trace metal redox proxies. And we grouped our 18 sites into, okay, I'll just see the legend there. So we binned them into redox, into 5 redox bins. And we're starting with 1. This is this pink color, which indicate the persistently euxinic sites. So these are the sites that have no oxygen in the water column, but hydrogen sulfide. And then it goes up to 5, and 5 indicates the least deoxygenated site, which is in this case the persistently euxinic ones, and everything in between are the numbers 2 to 4. And yeah, and this was based on the recent developments in the bottom water oxygen concentration based on the monitoring data where we had it available, or we used sediment geochemical data. We used enrichment factors to calculate the orthogenic enrichments of molybdenum and uranium, and that's a standard procedure to account for the detritoraction of the trace metals. And it also allows a rapid identification of differences in the orthogenic uranium and molybdenum concentrations among a large number of samples and study sites as we had it in our study. And here I show the results for the molybdenum and uranium enrichment factors across all our study sites, grouped into the redox bins. And this horizontal dashed line indicates the threshold above which we see the orthogenic enrichment. And we can nicely see here that basically all our sites have orthogenic enrichments, which is already like a great indicator for us that molybdenum and uranium work as redox indicators. And if we look now into further detail here, and I would like to draw your attention to comparing the medians of each of these bins that you can see here. So the medians are is the horizontal line that divides one box into an upper and lower box. For the molybdenum enrichment factor, that we can nicely differentiate between redox bins 3 to 5. So we have there the anoxic sites to the oxic sites. But we cannot really differentiate between redox bins 1 and 2. If we compare this to the uranium enrichment factor, we can differentiate between redox bins 1 and 2. But then between redox bins 3 to 5, it doesn't work so well. So this already shows us that molybdenum and uranium have some contrasting sensitivity here. And also that we cannot just fully rely here now on just looking on one of these proxies. So they cannot be used as standalone proxies because of their sensitivity problems. So the idea is you can either just plot them the way I did this, but in order to understand why they have the sensitivity limitations, you need to combine them. And therefore we tested the approach of Algae and Tuberculite from 2009, who brought the idea forward based on selective paths in modern open, marine and anoxic restricted deposition environments. And through this method, they identified 3 key covariation patterns of molybdenum and uranium, which they're linked to specific characteristics of deposition environments. And here I show now just all the data that I got from my own data set and also from literature data. And then I've overlain these covariation patterns that they identified. And the, okay, now it works. So the first one is this variation in the benthic redox condition. And that starts with the first increase of uranium. And that has something to do with that uranium is thought to occur at higher, that uranium, that the geochemical behavior of uranium is technically more sensitive than molybdenum. So uranium reduction occurs at higher oxidation reduction potentials than molybdenum. And that's basically what we see here, that uranium reacts first and then molybdenum. And that happens as conditions become more sulfidic, then we get this simultaneous increase in molybdenum and uranium. The other pattern that we see here is this manganese iron oxide shuttle. I will not go into so much greater detail here, but that's a mechanism by which molybdenum is very easily removed from the water column and transported to the sediment. And molybdenum is not and uranium is not affected by this. So that means we get a stronger increase in molybdenum in contrast to uranium. And the last one is the basin reservoir effect, and that's an effect that basically is restricted to euxinic settings. And that's again an effect that favors strong removal of molybdenum from the water column, because molybdenum then transforms into a very highly particular reactive form and is then being sequestered from by sulfides. And that goes so fast and rapid that the resupply cannot really account for that. And that's why we get this flattening on this curve. And again, uranium is not affected by this. So what we can actually see from this is that since this manganese iron shuttle mostly occurs in these green and light pink, so in these irregularly anoxic and hypoxic, so in redox pins 3 and 4, where we couldn't really see this nice differentiation of uranium. So that could be an identification that uranium, that's the reason why uranium is not so sensitive in our previous plots. And same applies for the basin reservoir effect, where molybdenum didn't really work. And yeah, so just as a summary for this, the principles of algae and tuberculite also work in our set of studies, in our set of study sites, and the particulate shuttling and basin reservoir effect can explain the sensitivity limitations that observed. And per definition, this particulate shuttling and the basin reservoir effect are not first order redox responses. So they're like secondary depositional factors. And that's, of course, very important to take under consideration, because that will mean that molybdenum and uranium are not only governed by the bottom water redox condition, but also by secondary factors. However, the limitations of the enrichment factors cannot all be explained by this cross plot that we just saw. Because if we think back to the first figure that I've showed, that uranium was also not working in redox bin 5. So we cannot really explain that. And that's why the goal of our second study was to better understand what other mechanisms and other secondary factors could be there that could explain that. So for this, we performed pool water analysis and trace mass signature extraction data for 3 contrasting sites. So they have different bottom water redox conditions. And we have Köyafjord as the most reducing site, where we have a typical redox donation. We have manganese iron shuttling here, but because the bottom waters are anoxic, we don't, all the oxides already being reduced, so we don't see a peak in the bottom, in the pool waters. Due to the high salinity, we have lots of sulfide reduction and release of hydrogen sulfide to the pool waters. In Gomafjord, we have high shuttling, and because the bottom waters are not anoxic, we get this nice increase of manganese and iron in the pore waters, but we don't see hydrogen sulfide because the sulfide reduction rates are just so low, at least up to 40 centimeter depth. And in Kovaha, we have an oxygenated water column, but due to very low salinity and very high organic matter input and the oxidation of methane from lower in the sediment, we have a high sulfide reduction rates and a shallowing of the sulfate methane transition zone. So even though Kovaha and Köyjefjord are very contrasting in the bottom water redox conditions, the processes in the sediment are actually relatively similar. I would just go here very brief. So we can also see the differences in the sequential extraction data. And we can just broadly see that mechanisms of enrichments of molybdenum here vary a lot depending on which location you're dealing with. And for Köyjefjord, we can see that molybdenum mostly increased in its very early stages of the extraction protocol. And at Gomafjord, we get this huge increase, huge enrichment of molybdenum in the top, which is but at depth, we don't see anything because of the missing sulfide. And at Kovaha, we have the strongest enrichment basically in this sulfide methane transition zone where we get the formation of pyrite. For uranium, we did similar analysis. I will not go into the details here because we see similar observations here that the enrichments are largely governed by the sulfide front in the sediment. So even though we have oxy conditions at Kovaha, we have basically higher enrichments of uranium than what we see, or similar enrichments to what we see at Gomafjord, which is technically more hypoxic. So to conclude here, that molybdenum and uranium enrichments are dependent also on manganese and iron shuttling and the sulfide front in a sediment. And that's of course particularly important if we want to assess the potential for future deoxygenation in marginal and non-reducing settings. Because that could also mean there could be indicators for, like early indicators for upcoming deoxygenation, which we don't yet see in the water column. Yeah, so these are just some take-home messages. Maybe I'll just leave it here because I'm running out of time. So, but maybe just one point I would like to make is that it's great that we're all collecting this oxygen data, but actually for us, it would be really great to get more sediment data for our proxies to validate them, because it was really hard to get that type of data. So yeah, with this, I would like to thank you, and I'm happy to answer your questions.
Okay, thank you. Unfortunately, time is over, but I invite everybody to approach you and make questions,
yes.
Also from the online people. Thank you very much. So now we invite our last speaker from this morning, Niels van Helmont, with his talk, Trace Metals and Their Use as Redox Proxy in Arabian Sea. He comes from the Utrecht University of the Netherlands.
Right. All right. Yeah, thanks. Well, good morning, everyone. Yes, one more talk about trace metals this morning. And I'm going to take you to Arabian Sea. I have to say this work, oh, sorry. I have to go back. I have to say that this work has been done by a PhD candidate in our group, and she's writing up the manuscript. She couldn't be here today, so I'll be presenting. So you may remember this slide from my GO2NE webinar in January. Sedimentary trace metals are a redox proxy, and that's because they're usually conservative in seawater, in oxic seawater, I have to say. Then they are present in the form of oxyanions, but in sediments they are enriched in the low oxygen conditions. And that's also what you see here in this cartoon. And the pathways of enrichment include association with organic matter, so that means absorption onto organic matter or in organic matter itself. Also simply diffusion into the sediment, and then there are sequential as oxygenic minerals. And they can also be shuttled with iron or manganese hydroxides, as Marijke also shortly mentioned already. And we see particularly strong enrichments under sulfidic conditions. So when a sulfide, we have the formation of oxygenic sulfide minerals. As promised, I'm taking you to the Arabian Sea today. So the Arabian Sea is one of the most pronounced oxygen minimum zones. So we know, of course, the Peru OMZ, but the Arabian Sea is definitely also on that list. So here we see a nice picture by Resplandi et al. 2012. And we see very high chlorophyll concentrations in the area. And that's because there's intense upwelling leading to high productivity. And then when we look into the water column itself, and here we see oxygen concentrations at 650 meters of water depth, and we see indeed that there's a huge area of this part of the ocean that is really devoid of oxygen. So the main research aims that we have are the Arabian Sea trace metal records providing additional insights in how these enrichments work, and as a proxy as well. And can we use these commonly used trace metals, such as molybdenum and uranium, as a redox proxy in this OMZ, which I have to say is mildly reducing, but I'll get back to that when we are going to the data. And also, how does the intensity of the present day Arabian Sea OMZ compare to the past? So our study area in the Arabian Sea is the Murray Ridge. That's a seamount in the Arabian Sea. And this was studied already before by a group of people, especially the paper by Kraut, I could recommend to read. But basically what we are looking at is we have 10 sites along the seamount. So really the top sites are where the seamount is impinging into the oxygen minimum zone, really the core of the oxygen minimum zone. And then we go down and we go slightly into oxic waters, and at depth, yeah, we can say it's oxic. And for the first results, I'm focusing on the two extremes. So the most anoxic site really in the core of the OMZ, and the one that's really below the OMZ, so the deepest site. See here is sediment data and pore water data, so solid phase, so really looking at sediment itself and the pore water from the interstitial water in the sediment. And so what we see here is that the organic carbon is really high in this anoxic site. Not a surprise, of course. The organic matter is just better preserved, and in this oxic site we see that the organic matter is much lower. Then we look to nitrate. We only see there's a little bit of nitrate in the bottom waters and a little bit in the surface sediments, but then it's gone. And actually, when we look at the oxic site, we see that this nitrate penetration is much deeper. It's almost 8 centimeters there. Then we go to manganese. There's no dissolved manganese in the anoxic part of the sediment. And when we go to the oxic site, we see that there's a lot of dissolved manganese actually. So that's a result of manganese oxides that are being used in the degradation of organic matter. And when we look at iron, then we see a lot of dissolved iron actually already starting quite shallow in this anoxic site, while we only see some iron coming up in the deepest part of this oxic part of the sediment. And then very interesting to mention, and that's also why I said it's a mildly reducing system, that's actually that we don't find any sulfide. And we've also looked at the characterization or the sequential forms of iron. We did a sequential extraction, and then we actually see there's little to no pyrite in these sediments. That's also, of course, also connected to the limited availability or actually no availability of sulfide. So that's very interesting. Then we go to the trace metals in more detail. So as said, no manganese in this anoxic site, and we also don't see manganese in the sediments. So I have to say this is dissolved manganese in the pore waters. This is manganese in the sediments, nothing present. And we do see actually a very straight profile for molybdenum in the pore waters. We do see also, a straight profile in molybdenum in the sediment. So it's just, a straight profile, but we do see it's higher than nothing. So there is some enrichment, if you could talk about enrichment. When we go to the oxic side, we see that the manganese is much higher, already relatively shallow, and we see a huge enrichment in the manganese in the sediments in the top. And these are manganese oxides. So because there's oxide available, these manganese oxides are still stable and not being used for getting metal degradation. And then when we look into the sediments, then we see a little peak here in the dissolved molybdenum, and that's actually some of the manganese oxides are starting to dissolve. So that's why we see this peak here. And we see also quite some enrichment in molybdenum. But this is the molybdenum that's actually absorbed on these manganese oxides. So that's really nicely visible in these sediments. Then when we go to uranium, it's a different story. Very interestingly, we see a very nice diffusion profile, and actually this really nicely mirrors the iron reduction, or the dissolved iron, which is a product of iron reduction. And what we see is just a diffusion that happens in this zone of iron reduction, and then we also can see at depth the sequestration of uranium. So that's a very nice example, and that actually fits with what we knew already. And when we go to this deeper oxic site, and then we see actually, indeed, at depth, we see some dissolved iron appearing, and we also see that uranium is decreasing, and also some enrichment in the sediment. So then when we go to all the stations and we put them all together in these nice bar plots, then we actually see that indeed manganese is increasing with depth. So in the low oxygen conditions, the manganese is not in the sediment, it's lost. But when the oxygen conditions increase, then we also see higher manganese. But for iron, we don't see a real clear pattern. So we only see some enrichment in the top side. But in general, iron is just showing flat profile. And that's also because, for instance, there's no sulfide, so there's no pyrite to be formed. So no enrichment in iron visible. Then when we go to the trace metals that we have looked at, so we've not only been looking at molybdenum and uranium, we've also looked at rhenium and vanadium, but also in other trace metals. But that's too much to show everything. But what we clearly see is indeed this nice enrichment in molybdenum, and then it decreases with increasing oxygen. And then at depth, we see these higher values again, and that's actually connected to manganese. And so this molybdenum is higher because it's absorbed on these manganese oxides are still stable under these oxygen-rich conditions, or more oxygenated conditions, I should say. And uranium has more spread, but we also can see that there is this gradient visible. For rhenium and vanadium, it's a little bit different. We do see a little bit of a trend in rhenium, but definitely not as clear as for molybdenum and uranium, I would say. And for vanadium, actually, we don't see a trend at all. So that would mean it's a little bit less, it's not applicable as redox proxy in this case. Then we compared our data with a nice review done by Bennett and Kentfield, who've been looking at euxinic systems, but also perennial OMZs, seasonal OMZs. So for instance, the Peruvian OMZ, but also the euxinic system, the Black Sea, for instance. And then actually what we see for most trace metals is that our concentrations are much lower most of the time. So molybdenum is lower, uranium is lower, rhenium is more or less on par, but vanadium is also clearly lower. So that's actually an interesting finding, that this Arabian Sea OMZ sediments are indeed showing a different picture, which might be related to this this low sulfide or no sulfide at all in the system. Then we also were interested, of course, in how it passed. So actually, we were lucky that there are already 2 published records of trace metals from this area in the Arabian Sea. So these NEOP cores, 463 and 464, which the data were published already back in 2006. And we now put our data next to them. And what we see then is actually that for molybdenum, we see in the OMZ that that the concentrations that we are finding are actually relatively low compared to what they were in the past. So we are here looking at thousands of years ago, so records of about 200,000 years ago. That's the deepest sites, but especially here in this part, we see much higher molybdenum concentrations, which leads to the conclusion that it must have been much more reducing back then. For uranium, we see we are in the same range as we were in the past, but we can also say, yeah, okay, uranium is maybe in that sense a little bit less sensitive, and we know that molybdenum is more connected also to sulfide. So that is probably the case. But what's also very interesting to note actually is when we compare our data from below the OMZ, then we see in the past really this huge spike. I would say, well, it's not a spike. I mean, it's 20,000 years in total, but clearly the OMZ was also wider back then. Huh? So it was clearly a more intense OMZ back then. So yeah, that brings me to the conclusions and the take-home messages. So despite of the absence of sulfide, molybdenum still appears to be the best redox proxy, at least for the system. And I think this is related to also organic matter. So molybdenum is also can be absorbed onto organic matter. We saw there's a organic matter gradient in the Arabian Sea. So this might be the explanation. Uranium measurements also follow the bottom order redox gradient, but the spread is much larger. So that makes it sometimes a little bit more hard to interpret. Rhenium and vanadium are only strongly enriched in the core of the OMZ, although we saw a little bit of a trend in rhenium, but it's definitely not as clear as for uranium and molybdenum. And when we compare it to other systems, it's clear that the Arabian Sea OMZ sediments are definitely less enriched than other systems, such as the Peruvian OMZ or the Black Sea. But yeah, then we are looking at really euxinic systems, so it's a different type of system. And the Arabian Sea OMZ was definitely wider and more reducing in the past. So yeah, that's where I would like to leave it. And thank you very much for your attention and organizing this very nice meeting.
Thank you. Time for questions? Oh, yes, please.
Hi, Niels. Thanks for a very nice talk. A couple of related questions about the core from below the OMZ. You could see that you had relatively high molybdenum aluminum ratios in these sites. But then, of course, when you showed the profile, influenced by the surface enrichment. So the question that follows from that is, what is, under the modern conditions, what's actually the burial rate or efficiency of molybdenum in these kind of locations?
Yeah, no, that's a very good question, of course. So are you referring to this one or just?
Yeah, this one shows it quite nicely. So the third profile here is molybdenum, aluminum in the site.
Yeah.
You can see the surface enrichment, and then immediately below it is very, very low values.
Yeah.
And then, so it implies that in the modern setting, you accumulate a lot at the sediment water interface, but that it doesn't actually bury permanently.
No, exactly.
But then how should we then interpret this step towards higher values at about 10,000 years?
Yeah, so that's actually a really good question. So I have to say there's a little bit of an uncertainty concerning the age model. So for the data, so that 10,000 years might also be 5,000 years, but still, I mean, the step is still there, so that doesn't really matter. So yeah, the top is really, yeah, we can relate the molybdenum that you see there to manganese. So there's still manganese oxides in the upper sediments, and then when those are dissolving or being reduced, then the molybdenum that's absorbed then goes. But then indeed, that step is indeed very interesting. So is that the source, or is that something else that at depth, maybe there is sulfide, because that's something that you could think. Huh? So the organic matter, the data that I showed is from the top 25 centimeters, but sulfate reduction might happen at deeper depths, right?
So it could be like a diagenetic overprint.
Exactly.
You don't interpret it as a primary paleoceanographic.
That could be. Yeah. That could be a diagenetic overprint. Yeah.
There is another.
Thank you for your talk. So when you say that the molybdenum is the best proxy redox, it's just in the Arabian Sea. So can we use the molybdenum as a proxy redox in other ecosystems like estuaries, for example?
Yeah. So based on what I showed here, indeed, molybdenum is working nicely. Huh? Yeah. And I think based on the whole literature, I mean, there's dozens, I mean, maybe thousands of papers even that use molybdenum as a proxy, and we do have to be careful because there are, you know, sedimentation rates, basin reservoir effect, all the factors that Marijke mentioned in the talk before, but all in all, I think we can conclude that molybdenum is a very nice proxy, and also in estuarine settings it can work. Actually, I've been authoring a study myself in relatively low saline conditions, where we also use molybdenum next to a suite of other proxies for redox conditions, and it also showed a very nice picture. It actually, so I would say yes.
Thank you. I was wondering what other hypotheses to explain what might have explained those wider or more reducing OMZ extent in the past?
Yeah, that's a very nice question, actually. So these are modulated actually by astronomical cycles. So yeah, that I guess I cannot go into detail, but I saw there's a talk this afternoon that also is dealing with the astronomical cycles. So we'll hear more about that. But basically, it's connected to the strength of the monsoons. So the more stronger the monsoon, the more upwelling, more productivity, and that's probably the case here. And so yeah. Then also there are also connections with Dansgaard-Oeser events and Heinrich events, and it's a bit complicated, but let's keep it that it's related to orbital forcing, of the planet. Yeah. Okay, thank you very much.
Okay, so it's the end for the morning. Yeah.
So, good afternoon, everyone. So let's continue from team of Lund University. I'm in postdoc at Lund University, and this talk is a part of my postdoc project about morphological variations of calcite microfossils, proton-based microcity to reveal the last 200 years of environmental changes in the Baltic Sea entrance. As you know, the Baltic Sea undergoes such as global warming, so ocean acidification. So in this study, I'm focused on the Baltic Sea entrance, especially the Danish Strait, which is a region as being vulnerable in the last 200 years due to combined hydrographic changes and human-induced impacts, such as eutrophication. That's why we need tools as sediment record and its contents of calcite microfossils to unravel recent environmental changes. For this, we use benthic foraminifera, which has noticed, and most of them have calcium carbonate shells. And foraminifera fossil records are designed to identify the appearance of human-induced impacts because most of the species have a specific ecology, and so it's easy to see differences in density and relative abundance. So the fauna has profoundly changed in the last 200 years in the Danish Strait, especially the subpolar species Elphidium clavatum. In the previous study of Chariot et al. 2019, they studied the foraminiferal fauna, and they did an H model with the sedimentation rate is between 1 and almost 6 millimeters per year, and this H model decreased with the depth. So here you have the different years, but you have to keep in mind the uncertainty. So here you can see in the industrial period, Elphidium clavatum was a minor species, and then in the great acceleration, these species show very large variations, and This is due to the hydrographic changes with the more sand inputs and also the increase of load nutrients in the strait. And then in the modern 21st century, this species decreased. So here, interested by the shell morphological variations of Elphidium clavatum sampled by Chariot et al. As a proxies of recent environmental changes. So we use synchrotron-based microcity to generate large 3D database, especially to look at the inter-individual variability to reach statistical valid conclusion. And we optimize post-data analysis, especially thickness and pore pattern. And then we generate 4D or 3D to all environmental changes. So we analyze 124 specimens at Spring 8 synchrotron in Japan, and we have between 5 to 10 specimens per depth. The analysis, we use two free software, SG, Fiji, and MeshLab. Image, you can see the raw 3D stack from the synchrotron microcity. Then segmented this image to divide the background from the shell, and from this binary 3D stack, we use Bonji plugin in ImageG Fiji to study the thickness of the entire shell. So you can see on this GIF, the blue colors means thinner shell and the yellow thicker shell. So this plugin is previously used in biomedical fields, and more recently, the shell morphometry of theropod was we use this method for foraminifera. Then we use MeshLab software to reconstruct shells. So here you have a panel of the 4 stages of preservation state of the shell, from perfect to well, medium, and poor preserved shell. So from this 3D reconstruction, we analyze geometric measure of the entire shell, so the external plus the internal walls of the shell. So we have access to volume, calcite surface, the surface volume ratio to look at the deformation of the. We also analyze the topological measures to determine the number of holes or pores of the entire shell. So we have the number of pores and the pore density, which is the ratio of the number of pores on the surface. So all the specimens were standardized by the same size ontogenetic effect related to growth rate. For the statistical analysis, we use non-parametric Mann-Kendall tests to look at the monotonic trends, and we use Kruskal-Wallis and Dunn-Poisson tests to differentiate specimens in between years. Concerning inter-individual variability, we hypothesize that a high inter-individual variability may reflect major changes in environmental conditions due to the diversification of calcification. And conversely, we hypothesize that a low inter-individual variability may reflect more stable environmental conditions. So now let's go to the results. So here you have the box plot with the age model, and in brackets there are the number of specimens scanned. All the values are adjusted between 0 and 1. So 0 means a thin shell and 1 thick shell. So here we found a significant trend over the last 200 years. And we also calculate the thickness loss about 30% in the 21st century compared to the 19th century. So in previous literacy, I only found one previous study of Fox et al. 2020, where they found a thickness loss of foraminifera in the Pacific Ocean between 76 and 20%. So our results fit in this range. So the loss of thickness seems to be global, but very specific to the species. And as you can see, around 2005, we found a thicker shell. So we don't know what's happened. So now when we look at the surface, around 0 round shape and flattened shape. So no trend was detected due to the large inter-individual variability since the 1960s. So since this date, we have more flattened shape, and these flattened shape are more adapted to low oxygen conditions because it's favor gas exchange through pore, and also flattened shape are favored by the thickness loss of the shell. And we can also notice in around 2005, we have more round shape and thicker shell. So now when we look at the number of pores, so 0 means low pores and 1 high number of pores. So here we found an increasing trend, and we calculate the number of pores increased by 100% in the 21st century, which is a lot compared to the 19th century. And here we can notice that there is the largest inter-individual variability in the 21st century. And according to a previous study of Conley et al., they demonstrate that the number and the duration of hypoxic events increase a lot in the 21st century in this region. And a high number of pores to low oxygen conditions. And now we look at the pore density. So no trend was detected due to the large inter-individual variability at different key periods. So early 19, early 20, at the beginning of the Great Acceleration, and in the 21st century. So interestingly, the ratio of the number of pores on the surface increased a lot, the variability compared to the number of pores, because we also noticed that the calcite surface increased a lot in the last 200 years. And these parameters also drives by the decrease in the thickness of the shell. So let's recap for 2005. So we have thicker shell with they are more round and with a lower number of pores. And so here we hypothesize that the environmental conditions improved in this area because there are several purification in this area. So to conclude, the 4D provide a baseline to reconstruct measure environmental changes in the Baltic Sea and France over the last 200 years. We improved the 3D post-data analysis, especially regarding thickness and pore pattern of the entire shell. And the main conclusion of this talk, we found a contrasting early 21st century trends because we observe major environmental changes over the last 200 with the thickness loss up to 30%, so acidification, and also a high increase of the number of pores up to 100%, so reflecting oxygenation also. So here you have example of the different shells found in this degraded condition. So these shells are thinner, more flattened with a higher number of pores and a higher pore density. And conversely, we also found a short improved environmental conditions. So foraminifera have a rapid resilience of the morphological variation of the specimen. So in this step, we found foraminifera with a thicker shell and a less number of pores and with a more rounded shape. So I would like to thank the financial support, the Swedish Research Council, FORMAS, the Carl Trigger Foundation, and Pages for the travel grant to come here. So thanks for listening.
Are there any questions? Questions? Yes. Always questions. Question is, how did you get samples from 200 years ago?
Sorry, could you repeat?
How did you get your samples from 1807?
Yeah, so from a core who was sampled in the Danish Strait. So the sediment core was sliced every centimeter depth. So the H model was performed on each layer, and the foraminifera was also from the different layer.
And one other question, wasn't there a major Baltic inflow in 2005? Is that what you were seeing of water from the North Sea?
Yeah, I'm still continuing to find. I think it's a combination of changes in hydrographic and also maybe sometimes a reducing of nutrient load in this area. But for the moment, it's quite difficult to find a very because it's not the precise year 2005. We have an uncertainty of 2 or 3 years. Sorry, I can't hear.
Any other questions? So what happened in 2005 and what happened after that it got wrong again?
We hypothesize that all the specimens used for the synchrotron were chosen randomly in each depth. So we assume that this specimen chosen reflects the main conditions in the different layer. So here maybe it can be a short improved of the environmental conditions, but it could be maybe also outlier or not representative of the data. We don't know.
Okay, thank you very much. So we can apply while we the next talk from Lelia Matos. She's going to talk about exploring cold water chromium isotopes as a proxy for intermediate water oxygenation state.
I carry this, right? I carry this. I'm not gonna. Yes, I'm not gonna. So good afternoon. Welcome everybody. I'm really happy to be here. I have a question. Sorry, if I use this here, the people at home don't. So then I should use it on the computer, or no? I know. Okay. Well, the normal way. So I'm very happy to be here to you. Unfortunately, our project got delayed, and what I'm giving you is preliminary data of our ongoing project. So this is a project under the MIT Portugal program. So I'm working together with Edward Boyle and Tiani Huang from the MIT. We are trying to see if we can use isotopes in cold water corals as a proxy for immediate water oxygenation conditions. It's the other way around, I'm sorry. Okay, so we had a couple of really great talks so far, and I don't explain to you that the oxygenation is occurring and is expecting to continue so in the next few decades. Projections show that. By now, you already know that models and projection need data to be improved, instrumental data, and by now you know everything there is to know about polysonographic data and how we can have it and why is it so important. So I'm going to skip this part. I had an example here for people that are usually not working in polysonography. This is a 1 million year record of SST that was taken from alkanones. So just for you to have an idea that how important polysonographic data is. Okay, also, by now you know all about polyoredox proxies, and some of them is used for redox conditions. Many of them, we've been hearing about them. I'm sorry for that. Today, I'm going to talk about chromium isotopes. For the people not so eased with chromium isotopes, a very simple explanation how this works. In the oceans come mostly from riverine input, and there's mainly two types of chromium species in the ocean, chromium-6 and chromium-3. Chromium-6 is highly abundant, it's soluble, it's toxic to most life forms. Chromium-3, it's bioavailable, insoluble, usually related to compounds. In certain conditions, chromium-3, and this is especially in areas that are rich in iron, that also could be emitted by bacteria close to ocean deficient zones. The reduced chromium-3 species is particle reactive, so it means it's binding to other particles that are falling to the seafloor and it's dragged to the seafloor. This is really important, especially related to ODZs, because usually ODZs are close to highly productive areas where many marine snow occurs. So you will have this big wide draw of chromium-3 to the seafloor. So if you combine everything, and if we know, I need to tell you also that when you have the chromium-6 being reduced to chromium-3, usually the lighter isotopes are reduced first. So chromium-3 is always lighter isotopically 6. So if we combine everything in places where we have the reduction from 6 to 3 and a wide draw to the seafloor of this chromium-3, that an increase of the isotopic signal on the remaining total chroming in the ocean. So you can see this here. It's a study made by Simon Bus, a postdoc from Ed Boyle. They measured chromium isotopes in seawater samples from the Northeast Pacific, close to the safe station. And what you see here, it's a depth profile. When you have a decrease in the concentration of oxygen, then you have a decrease in the chromium concentrations because of the chromium-3 to the deeper waters. And at the same time, you have a change of the isotopic signal to heavier values because ordolite values are being dragged to the deeper waters. So knowing this, a lot of researchers have been trying to see if this in foraminifera or tropical corals or other forms that then get preserved in the geological record. So we can use this as a paleoredox proxy. Especially in tropical corals, they found that there was an offset relative to the seawater on the skeletons of the corals. And they thought that this could be linked to the effects over the chromium-6 system that I'm not talking about, but there is some light effect, and also to the photosynthetic activity of the symbiotic algae that this have. So knowing all of this, we thought, well, cold-water corals are increasingly being used as archives studies. So why not try if they are a good archive also for chromium? So the advantage of using cold-water corals is that first, they don't have the symbiotic algae. So all this problem that we had with the tropical corals is not going to be seen. Some of the species, and especially the ones that I work reefs, and then they are really nicely preserved in the geological records. Also, they live at intermediate depths. I'm not putting that there, but that's really important for everyone that works with foraminifera and other archives worked in paleoceanography. One of the areas of the ocean usually really hard to reconstruct is intermediate, because you know the planktonic foraminifera are not that deep. The benthic foraminifera sometimes are too deep. So these corals that live around 5 to 1,500 meters water depth are a nice archive for these intermediate waters. So again, they are really nicely represented in the geological record. And if we do a long colony sampling, then we can even reconstruct decadal changes of the changes that happened when the coral was living. So the project, like I said, is to investigate if we can use the chromium isotopes from the corals to reconstruct redox conditions. For that, there are 2 steps. First of all, we need to find a way to establish a chromium analytical procedure to extract the chromium and analyze it from the corals. And once we are able to do that, then to compare it with the local water masses. So like I said, this is a collaboration between my lab and Ed Boyle's lab. And what I'm presenting you today is Ed Boyle's work. Okay, so they did this at the MIT. This study, we decided to compare 2 very different areas. So of course, we expect that if the system is working, the chromium system, that we will see this here in this area that is as a perennial upwelling and epoxic conditions, the Angola and Namibia margin. And then just to have like a control area, we decided to study another upwelling, but seasonal upwelling, where oxygen is high here on the Iberian margin, kind of as a control study. Okay, so the first part, including the development of the analytical procedure to extract chromium, decided to use Pertusa species because this is a species that is being widely used for paleoceanography. We already know quite some about it. And for that, of course, we need to clean the samples because of all the cleanings and the organic matter that sometimes it's there and may affect the signal. So when we analyzed the clean samples, of course, no surprise, we found very low amounts of chromium calcium, but that has been a problem. We had high blank effects on the measurements, and then we decided, let's increase the sample size because it's coral, so we can always have a B. We did that. We have metrics effects, too much calcium. So we are trying to overcome all these different problems that we've been facing. Of course, we have some data that I'm going to show you, but this is preliminary. So it's possible that having more samples than the values that we found may be shifting because some problems may have changed our signal. Anyway, we found a signature for the corals of -0.4. Here on the scale, you can see that the water masses for the Atlantic, the ones that are known and measured so far, are much heavier. So it happen either these waters do not reflect the waters close to our sample sites. We will measure water from the sample site, but we don't have it yet. Or there is a fractionation on the corals. Also, what we have for cold water corals is very similar to what they got for the tropical corals. But if there is fractionation, don't expect what is driving this fractionation is going to be the same because these corals live in very different conditions. So like I said, it's ongoing work. There's a lot to be done. We'll have to better establish this analytical procedure to extract the chromium and overcome all the problems that we've been having. And once we do that, we will compare it with the Lotema. If we are lucky and if we are able to find a way to correlate the chromium on the corals and the chromium on the environmental water, the local water, then we would like to have a case study and to reconstruct the redox conditions at the Namibia margin, what corals existed and they have been dated and we know that they were there in the Middle Holocene. It's expected that this changes in the occurrence of cold water corals in this area is related to the oxygen conditions. Like to test this, and at the same time we will compare it to the upwelling history since the upwelling is also driving this chromium system. So far that's what we have. Thank you very much for your attention. I would like to thanking all the funding agencies and everyone that collected and shared their samples with me, which was really important. Thanks. So any questions?
Yes, questions?
Hi, thanks for your talk. I was wondering whether you are planning to have a look at the speciation of chromium and its bonding environment in these corals, or whether you're just assuming that because it's corals and carbonates, it's all chromium-6.
I would expect the chromium in the corals to be the chromium-3 than 6.
Oh yeah, so yeah, but are you checking the speciation of?
As far as I know from the methodology that used, I think they change it all to just one species when they measure it. I'm not so sure, I'm sorry.
Okay.
It's exciting that you're working on Lophelia pertessa. It's like such a widespread coral in deep water all over the world. And it turns out the Angola ones are living in extraordinarily low oxygen compared to any other Lophelia recorded, I think. And so it'd be really, we have some low oxygen Lophelia off California, but it'd be really interesting to hear them around the world if you get a proxy available that can be used widely.
If you have Lophelia samples from California, that would be really interesting for the project, but actually for the North East Pacific, more information on the water chromium than in the Atlantic. So maybe we can talk later.
Okay, I don't have them, but I can direct you to.
Yep. Yeah.
So
that's great.
Thank you very much. Okay, I think we can catch up a little bit with time and go to the next one. Thank you very much. Can applause, please? About Milankovitch forcing on oxygen conditions in the Silurian.
Okay, yeah, thank you. First, I would like to acknowledge the FNRS and IES for funding this research, and my co-authors Anke-Stien from New Liège, David Vleeschouw from the University of Münster, Nicolas Sofbo and Anne Torstoy from the Geological Survey of Denmark, and Nicolas Thibault from the University of Copenhagen. So how do astronomical cycles modulate oxygenation conditions during the Silurian? Well, first of all, why would you study the Silurian? So I said the Silurian is going on and the climate was quite sensitive. So it's quite important to understand why the climate during the Silurian was so sensitive. And why would we use cyclostratigraphy to change in oxygenation conditions? Well, as some of you might know, the Earth's orbit, its axis, and the orientation of the axis are unstable, but vary with a regular period between 16,000 and 2.4 million years. And these cycles, when you combine them, you get something correlation curve, and this insulin, well, at certain a certain time of day, at certain latitude, maximum radiation during summer, and when it's at the minimum, actually the difference between radiation incoming during winter and summer is at its smallest. And actually this change in radiation actually impacts the climate quite a lot. And one of the examples is, for instance, ice during these times, during insolation minimum to lower summer temperatures in the northern hemisphere, leading to melting of the northern ice caps, which induce an ice. But during insolation maximum, summers were quite warm in the northern hemisphere and the ice caps melted. Another example of astronomical climate are the Mediterranean organic rich black layers of the Mediterranean basin. During insolation maxima, the African monsoon was enhanced, causing greater runoff into the Mediterranean basin, creating a freshwater layer, which cut off circulation, caused stagnation, which in turn caused anoxia to prevail in the Mediterranean basin. So how did the world sort of centered around the southern hemisphere with a large landmass around the southern pole and a smaller archipelago around the equator? My studied core is from here, just south of the equator, in a semi-enclosed basin south of the equator. Because our intertropical converter played quite a big role in modulating the climate, and there's also the monsoon, which in turn changed precipitation patterns and wind patterns during the year. Affect changes in oxygenation conditions? Well, usually in geology, there are two ways how you can induce anoxia: stagnation or by productivity. Well, during the productivity scenario, when the monsoon is really strong in a larger OMZ. Well, when the monsoon is weaker, you can actually have such a weak monsoon that overturning between the bottom and the top is so weak that the bottom water stays stagnant and become anoxic. So a strong monsoon during an insolation maxima would entail a productivity cycle. Oh, yeah, that's the next one. Yeah. So how can insolation affect either anoxia? When is high, you have lots of, and you can create a a bigger OMZ, lower seasonality, a sluggish circulation, and your bottom waters cannot get fully oxygenated, creating anoxia. In the Silurian, at my location, what happened? Well, for that, I first need to have a data of my core stress scanner, which gave me a multi-proxy data set with different elements, which can help me give a paleo-environmental interpretation of my data. Point 4, that's manganese. The dark red line is sulfur. So we can see that in one part of the core, manganese peaks, where sulfur peaks. And we know manganese primarily forms under oxic conditions or under suboxic, disoxic conditions. So within these lines, you can actually see a lot of variation, quite periodic. So the question is, what causes in my proxies? So one of the theories that it could be related to insolation or astronomical cycles. But for that, we need to do a spectral analysis. I did a spectral analysis on titanium over potassium because that proxy continuous record throughout my core to astronomical cycles. Titanium is we can use it as a proxy for coarse grain size. Well, potassium can be used as a proxy for clays. So when you have that ratio, when it's high, it means lots of coarse grain sized particles are going into my basin. When it's low, we have clays. And we know that if we want to have coarse grain sized particles in our basin, strong runoff. And to have a strong runoff, we need to have a strong monsoon. And because the monsoon is enhanced during insolation maxima, we can assume that titanium over potassium peaks during insolation maxima. So what we're seeing here is a so-called spectrogram of our cyclicity in depth with different observed orbital cycles. And all the variations you see going up and down, that's due to the changes. We can pick one cycle. We know how much time we have, so we can convert from the time domain. When we do that, we can see a lot more over the data, and we can look at how, as said, if titanium over potassium peaks, maximum manganese anoxic proxy and sulfur anoxic proxy. And we can look at the phase between those two, and we see that sulfur peaks when titanium over is the minimum, which indicates to me anoxia proxies. And we can actually see that all the anoxia proxies are opposite to the grain size proxies, which confirms my interpretation that anoxia in the basin was caused by stagnation due to forcing. So data set. The first is during insolation minima, during peak anoxia conditions in the basin. So during our productivity season, we have some productivity, organic matter that goes down, but then we have a winter monsoon mixing season that properly mix, and the basin bottom stays anoxic. While during a insolation maxima, during peak oxic times, we still have a productivity season, some organic matter gets produced, and oxygen levels go down. But when we are again in a mixing season, our basin gets fully mixed. gets fully mixed, overturned to an oxic state within a basin. So from our observations, from our results and observations, we can conclude that astronomical cycles play a pivotal role in modulating oxygen during the acceleration. Yeah. So
thank you very much. So are there any questions?
Thank you. The sites that you're looking at, are they coastal mostly, or they were coastal? When you say mixing is bringing oxygen, it means it's on the shelf, right?
It's the deposition environment, not more than 350 meters depth, according to literature. So we're quite a shallow basin.
Impact the oxygen minimum zone like at depth?
Well, as I said, the base not fully connected to the open ocean. So what happens in the larger Syrian ocean cannot be extrapolated from my results.
And I have a final question. Can you go back to the slide where you were showing the phase diagram? Like I really see these in phase between the oxic and anoxic thing, but I'm sorry, can you talk about the other side of the slide where I'm supposed to see the link with the astronomical parameters?
Yeah, so since we have here, we filtered out one of the cycles in blue and same period. So one of the many orbital cycles, this is the 405. When we look at it, we can see this one. Well, here it peaks, it's the minimum, it's at the minimum. So the phase is different. So when one goes up, the other goes down. You take out one of the orbital cycles. So because it's like I said, the insolation signal is a composite of many orbital cycles, and you can, by using some fancy mathematics, you can pick them all out and then compare them to each other. This can give insights into the paleo-environmental setting.
Thank you. We have time for one more question, maybe.
To sort of support your chronologies?
Well, you always have biostratigraphy, which can be used to infer how much time there roughly is, so plus or minus a few million years. And there are also a few bentonite beds, so uranium lead dates, which also further constrain your how much. But from the orbital cycles, you can infer how much time there is by looking at the relationship between the different cycles present.
And now we will continue the session with Nathalie Fagel.
Okay, thank you. Okay, so we continue the session. The talk will be given by Vyacheslav Kohn, and he will discuss about the effect of the Panama seawater closure on the Pacific oxygen minimum zone during the Pliocene.
Today, I would like to show use our preliminary results on of the Pliocene Panama Seaway closure on the tropical Pacific oxygen minimum zone from model simulation project. and with collaboration with Kiel University and GeoMar. So first, let me give you some brief introduction. So for the modern day, the world's largest oxygen minimum zone resides in the eastern tropical Pacific. Ocean ventilation coexists with production. So this shadow area is at the periphery of the is under influence of the more cyclonic circulation, preventing subduction of oxygen-rich water. So on the other hand, the modern continental geometry, including the Panama Isthmus, shapes the local regional climate and marine biogeochemistry in this area. So it's is of interest to investigate tectonic transition occurred during the Pliocene with opening and closure of Panama Isthmus. As I already said, this work is carried out the Fargo project, the warming world, and I'm a person is in charge of modeling part of this project. So the modeling part focusing on the study of evolution of the tropical ocean circulation and oxygen minimum zone across warm paleoclimates using two global climate models. So we employ Kiel climate model and Hadley Centers model in biogeochemical model PISCAS. So we will be focusing on the three key Pliocene interval where closing Central American Seaway occurred, and another period of interest is the mid-Pliocene warm period, and finally last 800,000 years, including interglacial intervals. So first, very briefly about the Kiel climate model. This climate model consists of atmospheric part, which is ACAM 5, atmospheric general circulation model, coupled with ocean model NEMO, with interactive sea ice scheme. So this model was widely used for many paleoclimate modeling studies. In particular, we studied the sensitivity of the hydrological cycle, interglacials. Also, we studied Holocene evolution of climate and marine biogeochemistry in the tropical Pacific, and variability for the Pliocene simulations with open and closed Central America seaway. So this table summarizes the series of model simulations we are planning to perform within this project. So we are focusing on three key paleo intervals. I already mentioned here the Miocene-Pliocene interval. So this talk will be focusing on this period. So here we employed 3 simulations with different depths of open sun wave corresponding time interval within this period. Then from climate model will be used to force marine biogeochemistry model Piscus in offline mode. This slide demonstrates the experimental design. So here you American Sea Wave with Panama Isthmus and just replaced Panama Isthmus, so land 3 land grids in the ocean model replaced by ocean Central America, open Central America Sea Wave is different. So here's intermediate depth, and here is the shallow depth. So it's obviously that the removing of the land grids North and South America allows for the water mass exchange between this plot shows the vertical cross section across the Central American Sea Wave. For the meridional velocity and positive values corresponds to northward motion from Pacific to Atlantic, and negative from Atlantic to Pacific Ocean. So the main result here that the measure through the open Central American Sea Wave is directed from Pacific to Atlantic. So this major part is outflow from Pacific to Atlantic, and this outflow is driven by interbasin sea level gradient because the Pacific has a higher sea level compared to Atlantic, and this creates a pressure gradient force. There is only a surface layer where the velocity controlled by seasonal variations in the trade winds, and also possible the counter opposite direction of currents from Atlantic to Pacific. But the outflow consists of 14 sphere loop and only 1 sphere loop from Atlantic to Pacific. So what is the main consequence of the opening of the Central American Sea Wave? This is input, also outflow of towards the northern Atlantic creates low salinity anomalies here, which leads to the slowdown Atlantic meridional overturning circulations. So you can see here that for the control simulation with closed Panama, it's about 14 sphere loop, while for the open sea wave is about 11 sphere root. So this is because of freshening. The opening creates fresh water mass in the North Atlantic, northern part of Atlantic. And this slowdown of Atlantic meridional vertical circulation in turn creates a typical pattern in the sea surface temperature with, in general, cooling in the northern hemisphere and the warming in the southern hemisphere, because Atlantic meridional vertical circulation brings warm water from southern hemisphere to the northern hemisphere, and the slowdown leads to the opposite. So it's more water accumulate in the southern part, and so there is a between two hemispheres. And if you look at the global redistribution of water mass transport when opening the Central American Sea Wave, then we can see that this additional 13 sphere groups flowing from Pacific into the Atlantic Ocean is balanced with reduced outflow through the Indian between Indian and Pacific Ocean, and between Southern and Pacific Ocean. So then after entering Atlantic Ocean, this amount mainly transported towards the southward, and this is creates a strengthening of southward Brazil current here. So this extra 12 sphere groups now entering the Southern Ocean, and then promote the intensification of circumpolar Antarctic current. So shown here. So very briefly, just to introduce the main factor for the ocean for the supplying oxygen-rich water. So because this shadow area and affected by cyclonic circulation, the main main source of oxygen-rich water is a subtropical anticyclonic circulation, which creates subduction of oxygen-rich water. Then this water travels within the thermocline towards the eastern equatorial Pacific here. And on the other hand, this wind system creates the trade winds, which promote the upwelling, strong upwelling in this area with high biological production. So this is a mechanism of the oxygen minimum zone in this area. So according to our model results, the opening of Central American Sea Wave drives large scale eastward subsurface current. So the opening associated with a stronger eastward current, and this in turn leads to the better ventilation in this area. So the supply of oxygen intensified, and then this leads to more oxygenated conditions in this area. So what we see in our experiment, Central American Sea Wave is is the high oxygen concentration compared to the closed Panama Seaway, and this is accompanied with changes, corresponding changes in water age. So the water mass becomes younger, and the oxygen concentration is higher. And another factor is important factor is biological production. In our preliminary results from our model simulations, we found that the net primary production decreases in the experiment with open Central American sea wave. So here is a negative anomalies. And this negative anomalies in net primary production is associated with the export of nutrients from the Pacific towards the Atlantic. So now let me conclude. Yeah. So we performed a set of model simulations with the character climate model driven by different bathymetry corresponding to deep or shallow Central American Seaway, and net water mass transport through the open seaway directed from the Pacific into Atlantic and driven by interbasin sea level gradient. So the Pliocene closure, Central American Seaway closure, has led to termination of fresh water supply to the North Atlantic from the Pacific, and therefore to intensification of Atlantic Mediterranean circulation. It was found that the open seaway is associated with large-scale eastward subsurface flow in this Central Pacific, thereby enhancing water mass ventilation and marine oxygen concentration. And model results show that opening is associated with the export of nutrients from the Pacific to Atlantic. This leads to reduced NPP. And important that both factors, altered net primary production and ocean ventilation, contribute both to the oxygen minimum zone intensification in the Central Pacific, when assuming modern day configuration with closed Central Mexico. Yes, thank you very much.
Thank you. Is there a short question in the room? Yes, please.
Thank you very much. About a decade or two ago, there was a big debate whether this westward flow or eastward flow happened. So based on the Mayaurima experiments, and Dornoff had a theoretical argument that the flow should be from the Atlantic to the Pacific, and models are wrong because they have too much mixing. Is that resolved?
Second.
So I think your model was...
You mean this
yes,
interbasin exchange?
Yeah.
According to our results, because of high sea level in the Pacific.
Yes, that's what was Mayaurima found 1990. But that's because of the different salinity values basically in the basins. And after exchange, after the strait is open, that equilibrates to some extent. And then Dornoff said it in the end, in equilibrium, it's the other way around. But I haven't paid attention at all to that for the last 15 years, probably. So is it this debate, or there was a big debate then? Is that settled? Not, maybe it is.
In our simulations, this is our result. So we have only in the surface layer where the wind driven the surface layer driven by wind But no, okay, we have another experiment with the deeper sea wave. Then we can see opposite direction. I can just a second if you I just when we have a deeper, then there is a opposite inflow from the Atlantic to the Pacific. Yeah, this is because effect of the difference in the density, vertical distribution of density. It may happen. Yeah.
Okay. Okay, thank you. So we move to the next talk. So the person is not present physically here, but she will give the talk. I assume she's there. So her name is Catherine Davis.
My name is Catherine Davis, and thank you so much for taking some time to watch my talk. I'm really sorry that I can't be there.
Okay, you can start.
In person, but I hope that I'm able to interact with many of you over the course of this next week. So today I would like to talk a little bit about some work that I've been doing over the past year looking at the deglacial shoaling of the eastern equatorial Pacific oxygen minimum zone. I think most of us are likely aware of expansions of the oxygen minimum zone over recent decades. I'm showing here one classic example from the tropical Pacific with depth on the y-axis and time running into the aughts along the x-axis. And the blue color here indicates where the oxygen minimum zone is in the water column. And as many of us are working on understanding these changes and their effects currently or projecting into the future, one of the questions that I'm really interested in is, what has happened in the past and how can that help inform our oxygen future? One of the most recent examples of past climate change is the last deglaciation. And we do know that oxygen minimum zones globally expanded during this deglaciation, especially during periods of rapid warming. Some of the most extensive evidence for this comes from the North and South American margins, and I'm showing here on the right one example of a synthesis of benthic records from the Mexican margin. So more red here indicates a greater expanse of oxygen minimum zones in contact with the shelf, with more red indicating a vertically expanded oxygen minimum zone. We see this expansion especially during the Younger Dryas, which was one of the major pulses of deglacial warming, as well as the early Holocene, when the Earth warmed to a climate similar to pre-industrial. Now, there's a reason why this synthesis focuses on the shelf, which is that's where we have most of our proxies for paleo-oxygen minimum zones. And that means that important oxygen minimum zone regions, such as the eastern equatorial Pacific, with much less impingement of the oxygen minimum zone into benthic environments, have been a little bit more opaque. And that's really where I would like to take us today. The eastern tropical Pacific is the site of the largest expanse of dysoxic waters in the modern ocean. And to get us oriented, I'm showing here just a map of oxygen in milliliters per liter at 200 meters depth on top. And then we can also look at an artificial section through the same region, going here from 10 degrees south to 10 degrees north. And in the modern oxygen minimum zone, we can see that that extends from just about 100 meters down to around 1,200 meters or so. And then below that, oxygen increases again. Now, this was not always the case, and there's actually a fairly robust body of evidence that shows that in the last glacial maximum, there was a massive body of low oxygen high somewhere around here. But there are far fewer records that let us get at that mid-water oxygen minimum zone region above. So really, the question that I would like to ask today is, how do we go from a state that looked like this in the last deglacial to one that looked like this in the modern? And to do this, I'm actually going to focus on this core that's shown here as a star, TR16323. It does lay quite a bit below the region that we're interested in the water column, but has a magnificent fossil record. Before we get into that, though, let's start with what we do know. And this is admittedly quite a small and selective group of records available from the eastern equatorial Pacific that I'm showing you, but it should allow us to get a quick summary. And I will also say that for those of you who also love to look at marine oxygen proxies, this is really a slide for you. For everyone else, hopefully I can give you the gist here real quick. So what we're looking at is the modern conditions on the left of this plot and conditions in the last glacial maximum on the right of this plot. The top panel here is looking at iodine-calcium ratios of a thermocline-dwelling planktic foraminifera from one site in the eastern equatorial Pacific. So here, what we see is indication of exposure to low oxygen waters when iodine and calcium ratios are particularly low. So here, that's through much of the late glacial interval, and then especially in the peak deglaciation. Below that are a collection of some organic carbon records, which suggest a peak in productivity right there in the peak deglacial, maybe coinciding with low subsurface oxygen as well. Below that, the bulk nitrogen isotopes tend to give us a steep step change from the glacial to deglacial conditions, and this indicates either a decrease in oxygenation or maybe a change in nutrient conditions. And then finally, we have the nitrogen isotopes from some thermocline-dwelling foraminifera, which might suggest a peak in denitrification in the thermocline right around the deglaciation. So overall, there's a good indication that what we were seeing were some clear changes in midwater oxygenation associated with the deglacial interval of climate change. But there's not quite a clear story here yet. So I wanted to get at this from a little bit of a different angle, using the planktic species Globiortiloides hexagonus. These guys are a foraminifera, so a shelled protist, that have an excellent fossil record. They appear to be endemic to the oxygen minimum zone, so what I'm showing here is a pie plot from some previous work that we've done. And what we found was that when you look at the oxygen minimum zone from the eastern tropical North Pacific, 87% of living foraminifera from the oxygen minimum zone are composed of this species, hexagonus, shown in yellow here. And that really opens up a lot of potential to apply some paleontological and geochemical tools to these problems using their fossil shells. We actually found one other really interesting thing from this previous work, which is that the porosity, shell size, and average chamber number associated with the final whorl all decrease with increasing oxygenation in these toe-retrieved modern foraminifera. None of these relationships are linear, but they all show a clear and statistically significant trend. So the first thing we wanted to do at our down core site was to use this morphological trends that we found to test whether hexagonus actually maintained an oxygen minimum zone habitat in the eastern equatorial Pacific analogous to today through the deglaciation. So what we're looking at here is a series of violin plots of individual hexagonus morphology through time since the last glacial maximum. Based on what I just showed you a moment ago, we would expect that a population of hexagonus that was living in, for instance, more oxygen-rich waters would have smaller shells, fewer chambers in the final whorl, and a lower porosity. I've plotted each of these traits in individual shells through time, so essentially if we saw a shift in the population in the downward direction on each of these metrics, we would suspect a more oxygenated habitat. And the truth is, this is quite an ugly plot because the punchline is there is not much of a trend. There are a few shifts in morphology, but there is no systematic change. So from this, we can conclude that the environment that hexagonus was living in has really not changed much from modern distributions. They're likely tracking an oxygen minimum zone habitat through time. So now I would like to spend the rest of my talk comparing the oxygen and carbon isotopes of hexagonus to those of some other foraminiferous species to look at changes in habitat and therefore changes in oxygen minimum zone depth. To illustrate what I'll be looking at, I'm showing an example of an oxygen and a temperature profile in white and blue here, as well as a theoretical oxygen isotope curve in pink. And this should illustrate what we know about where we would find these three species of planktic foraminifera and what their oxygen isotope value should be. So first we have hexagonus, which is down in the oxygen minimum zone, and this should have the highest oxygen isotope values. We can compare that to records from these two other species. So we have deutetrae, which lives in the upper thermocline right here, likely tracking the chlorophyll maximum. And then finally, we have ruber, which lives up here, quite close to the surface and the mixed layer, with relatively low oxygen isotope values. And the relative changes then and the oxygen isotope values between these species records should tell us something about the relative positioning of habitats in the water column. So let's get into that. What I'm showing here are now the oxygen isotope records of Hexagonus as well as these other two planktic species from previously published data, and a benthic species, Sibicidoides willistorfi, in blue at the bottom. So here we have Ruber on the top with the lowest oxygen isotope records, then Deutetrae in gray, and Hexagonus in yellow. And where I'd really like you to keep your eye on in this plot is the difference between Hexagonus in yellow and Deutetrae in gray. So below this, I'm now going to add the carbon isotopes from the same shells. And here I would like to especially call your attention to, again, Hexagonus in yellow, but in this case, Sibicidoides willistorfi are benthic species in blue. Now, there are a ton of things that can impact foraminiferal carbon isotopes, but the key thing to keep in mind here is that both of these two species are probably recording delta C13 very near equilibrium with the carbon isotopes of dissolved inorganic carbon. So this is going to tell us something about the carbon levels in source in their immediate calcification environment. So now let me walk through this plot and hopefully tell you a little bit of a story about what happened in the eastern equatorial Pacific oxygen minimum zone. So at the end of the last deglaciation, it looks like the oxygen minimum zone was sitting much deeper in the water column than the thermocline. And to get at this, I'm mainly looking at the difference in oxygen isotope records between Deutetrae and Hexagonus in this top plot there. A shoaling appears to have occurred beginning around Heinrich Stadial 1 and continuing up into the Bowling-Allerod, right at the end of this blue stripe here. And meanwhile, in the carbon isotopes, we see an incredible similarity between Hexagonus and willistorfi. And this indicates that they're probably calcifying in a very similar carbon environment. So altogether, these records support a late glacial to early deglacial condition with a very deep oxycline extending at least to the benthos and probably connecting with a deep glacial carbon reservoir. When we get into the peak deglaciation, this extends from about the beginning of the Bowling-Allerod to the Holocene onset. We see a dramatic shoaling of the oxygen minimum zone, with oxygen isotope values of Hexagonus basically converging with those of Deutetrae and the thermocline. At the same time, we see a continued similarity between the carbon isotopes of Hexagonus and Sibicidoides. We're interpreting this as a maintained connectivity of the oxygen minimum zone and this deep carbon and low oxygen reservoir, but with both having shoaled to now fill a massive block of the water column. This would also be occurring coincident with and consistent with evidence for reduced stratification in the region, as well as increased productivity and potentially greater denitrification, especially in the thermocline, some of the evidence for which I mentioned earlier. And then finally, we get into the Holocene, and our Hexagonus isotope record starts to demonstrate a more modern condition, with a shallow oxygen minimum zone relative to the thermocline. So in this case, I'm looking at a relatively small gradient between Hexagonus and Deutetrae records, although certainly much larger than we saw in the peak deglaciation. At the same time, we're starting to finally see a separation of the carbon isotope values between our pelagic Hexagonus records and benthic Sibicidoides. And this is occurring at the same time as we know that the benthos has become better oxygenated and more carbon poor. So altogether, I would argue that adding records from within this pelagic oxygen minimum zone environment allow us to piece together a fairly coherent picture of what was happening in the region through the last deglaciation. I think critically, our results suggest a really dramatic shoaling of the eastern equatorial Pacific oxygen minimum zone during the last deglaciation. And if our interpretation of connectivity with the deep reservoir in this period holds, it supports that the eastern equatorial Pacific was probably a key region in directly venting this stored carbon to the atmosphere. Finally, this interpretation would imply that at least in the eastern equatorial Pacific, we're not just seeing a contraction and expansion of the oxygen minimum zone with climate change, but we can see a fundamental restructuring of where carbon and oxygen are stored in the water column. And finally, I think that these shells of Hexagonus are likely to be an archive of other information about conditions within the oxygen minimum zone. I actually mentioned strontium to calcium ratios in my abstract, and unfortunately, while I didn't quite have time to get into that today, it's something I would love to talk about. So please do feel free to reach out to me about those preliminary results, as well as any other questions you might have about the talk. Again, thank you so much for your attention.
So we move to the last talk of this session. This talk will be given virtually by Johannes Spain, I think. Hi.
Hi, thank you very much. Can I start?
Yes, I think it's fine. It's okay. Thank you.
Welcome everybody to my talk, Stratification, Sedimentation, and Hypoxia in the Coastal Environment, Model-Based Assessment and Implications for Management, also on behalf of my co-authors from Helmholtz-Zentrum Geesthacht, Joanna Staneva, Ute Dewe, and Corinna Schrum. Now I would like to go back with the slides. Maybe someone can help me to one slide back. I cannot. This was forward actually. Okay, good. This is the outline. I will compare two coastal systems, a non-tidal one and a tidal one. And the talk is about the dynamical characteristics, how the nutrient loading and turnover works, how this affects oxygen conditions. And I want to conclude with some common issues and differences between the two systems and how this informs possible management solutions. For the modeling of the coastal environment, it is necessary or very helpful to do a model nest. For this, we use usually CMEMS product, at least for the physical forcing of the open boundaries. Here, I apply this to two coastal systems, the Elbe Estuary and the Odra or Chichichin Lagoon. And for the ecological boundary conditions, I use usually modeling data from a larger ecological model or also observations. Observations are very important for the prescription of the land boundaries, such as river discharge, nutrients, oxygen, and so on. The modeling system is a schism-farben eCosmo. This schism is a hydrodynamic community model that involves many modules, such as sediments and so on. Here I use just the hydrodynamic part and couple this to eCosmo, which is a geological model for the lower trophic levels, via the FABM, which is the coupling interface between hydrodynamic and ecological models. The FABM is a lower trophic, involves the lower trophic levels, such as three functional groups of primary producers, two functional groups of zooplankton, nutrients, oxygen, and also sinking tracers. This is the ditrid, opal, and it is able to store actually the sinking tracers as nutrient pools in the sedimentary layer, which is quite important for some coastal systems. I start with the Odra case. Here I use the modeling framework, setting up a simulation, Baroclinics 3-dimensional simulation of physics and ecology, covering the period of 2012 to 2013. The model is driven by CMEMS 3 analysis on the physical side and eCosmo simulations presented by Dewe and Schrum 2013 on the ecological side. And also atmospheric forcing from German Weather Service and observational data for the forcing of the Odra River and the small contributaries. The model has been validated against observed sea surface temperature and sea surface salinity. I show the Taylor diagrams on the right-hand side. You can see that errors are quite small, especially for temperature, but correlation is very good for temperature and acceptable or better for surface salinity. This is important to tackle these dynamics because the salinity and temperature are the drivers of stratification, especially in the small straits connecting the Odra Lagoon and the Baltic proper. The next part of the validation concerns the oxygen and the dissolved inorganic nitrogen dynamics. Yeah, you see the Taylor diagrams on the right. Good correlations for both oxygen and nitrogen. Errors are acceptable for dissolved inorganic nitrogen for most of the basins. The same for oxygen, with the exception of the domain on the northeastern part. This is something still to be improved. And very important to tackle the system dynamics is actually the primary production in the shallow waters of the Odra Lagoon. This is shown on the left-hand side. The two top panels show observations. These are the circs against model results. This are the solid line. And below, the same for nitrate. Important is that nitrate is actually depleted at some time between spring and summer. And this is shown in both observations and model results. Yeah, the basic functioning of the system is we have high primary production in the shallow waters of the Odra Lagoon. There also we see growth of zooplankton. Both produce detritus or particulate organic nitrogen, which sinks down and is remineralized either in the water column or at the bottom. And this will lead to some degree of oxygen depletion. The oxygen depletion is known to be very high at the bottom of the Baltic. This is also seen from the model results. In the shallow waters of the Odra Lagoon, this is usually not the case because the shallow waters are usually well mixed, but we see that in the deeper waters, like in the navigational channel connecting Szczecin to the Baltic proper, we see some degree of oxygen depletion. Yeah, I talked about stratification. Here's one example. The station in the Peene current. It shows very low degrees of salinity during summer, but rising temperature. So salinity is actually the cyan-colored line. Temperature is a red-colored line. And at the same time, the potential energy anomaly has also maximum values. It moves away from the bottom line. This is the blue line, and this means we have temperature enhanced stratification in this Peene current in summer. In the same position, we see production of ammonium at the bottom and low oxygen values. And yeah, to go a little bit more into the detail of the coupling between physics and ecology here, I showed this time series at the same station where we see low values of diffusivity, lowest panel during summer. We see peaks of summer temperature that are associated with peaks of ammonium production at the bottom, so peaking ammonium concentrations at the bottom, and minima of the dissolved oxygen saturation. Yeah, what is interesting about the inner part of the Odra Lagoon is that the two parts, the small lagoon and the great or large lagoon, they are actually to some degree, they show decoupling. With quite different salinity and concentrations between the eastern part and the western part, and the same applies to organic, to biological traces or biochemical traces such as nitrate. And yeah, this is well known that there is a degree of decoupling between these two parts of the basin, and actually that neutral reduction that works for the Odra does not well affect the western part of the lagoon. And this model can actually show a little bit how this is generated. Some older maps of the area imply that the coasts have looked quite different in the past. It's however quite difficult to reconstruct the details. What is sure is that the navigational channel we have today was not there in the past, and by the construction of this channel, the Oder River waters are routed more quickly to the Baltic, and they cannot well mix within the lagoon as a whole. This can be seen, for example, from a water age tracer, which is shown on the bottom right, where the youngest water is at the mouth of the Oder River when it enters the lagoon near Stettin, and ages increase not so quickly towards the Baltic, but quite strongly towards the western part of the lagoon, that is the small lagoon, and beyond. This shows that there is increased residence time in this part, and this shows also the degree of decoupling of these waters. Also, we see the navigational channel shows increased summer stratification. This is a left picture on the bottom, and low oxygen values. And with this, I come to the Elbe system. This is quite different because it's driven by tides. We use the same model for the system. I speed a little bit up here. The model has been validated against tide gauges, velocity measurements, and biogeochemical observations. Everything is described in more detail in this publication. Cited is shown on the bottom. I won't go into details here. What's important about the system, it has not so much primary production on its own, but it receives very high organic loads from the eutrophied Elbe River. And these high organic loads, they break down upon reaching the deeper waters of the port of Hamburg and beyond. And there starts an intense biogenic particle production of detritus, phytodetritus, or particulate organic nitrogen, which sinks to the ground and is somehow trapped on spot. Why is it so? Here you can see more details to this. You see high concentrations of nitrogen in the bottom layer, in the port basins, top left panel. At the same spots, you see high ammonium concentrations and oxygen depletion. And looking at the time series of a number of biogeochemical variables in connection with temperature and stratification, we can see again that the stratification and temperature maxima coincide together with high values of ammonium and low values of oxygen. Very similar to what we saw in the Oder system in the Peene current. It is well known that the Elbe system is a massively engineered system. So since medieval times, there has happened diking of the river, building of new channels, removal of old channels, and finally there happened the deepening of the main channel. In the old system, we already had two branches, but actually the southern branch, which was the more important one, was a meandering channel, and all the system has been somehow simplified and straightened, and one has cut off the meandering parts more or less. In the system today, we see a prominent barometric jump between the shallow upper tidal river and the deepened tidal river between that includes the port of Hamburg and the Elbe estuary. In this location, we see a strong flood dominance driving every sinking tracer upstream. And in the shallow part, it's the opposite. We see abdominance driving every sinking tracer downstream. In this way, sediments and including sinking organic particulates such as detritus are trapped in this area. Due to the barometric jump, we have a partial reflection of the tide, and which involves a weakened ebb current. And this weakened ebb current induces temperature stratification in the water column, which cannot be mixed up quickly. So there's a persistent periodic stratification in this area, and this makes it more difficult for oxygen to reach the bottom. How can we prove that this mechanism is into play? We manipulate the model and remove the influence of temperature onto the equation of state and compare such a run with the previous run. And we see that in the difference, the part of the organic sediments leaves the port area. In the same place, we see lower ammonium concentrations, higher oxygen, and higher nitrate concentrations, which shows the system is moved away from oxygen depletion, and at the same time, however, we lose some ability to denitrify, which then leads us to the question of trade-offs.
You conclude?
Yeah, I can conclude. I just try to come to my last slide. Yeah, here's I come to my conclusions. The coupled physical ecological model reproduces the dominant physical and biogeochemical processes in the Outer Lagoon Elbe estuary. Both systems have been subject to massive human intervention, and both systems suffer from eutrophication. Clear differences arise from the presence of tides in the Elbe, actually, that drive redistribution and trapping of organic sediment particulates. However, and I couldn't show this today, but I think the presentation is stored and you can check it later. Tides can be useful actually to improve mixing and reoxygenation, especially considering changing the channel geometry in such a way that this is favored. In a non-tidal system like the Outer Lagoon, nutrient reduction is actually imperative, but increasing the connectivity, that means connecting parts that are now decoupled from the basin better to the main river might support such an effort. Thank you for your attention.
Thank you.
All right, so maybe we'll go ahead and get started. Welcome to our third session today on microorganisms and microbial processes associated with ocean oxygen and deoxygenation. I'm honored to introduce our keynote speaker, Professor Don Canfield from the University of Southern Denmark. Don is the director of the Nordic Center for Earth Evolution there. He's a member of the American National Academy of Sciences, a member of the Royal Societies in Sweden and Denmark, and a member of the Royal Society of London, I think. So Don has also written the book on oxygen, A 4 Billion Year History, and I encourage everyone to check it out. And in the meantime, I'll turn the floor over to Don.
Well, thanks, Sean. We're way too many royals there. I hope everybody is caffeinated and ready to go, because you're going to need it as I through the details of the nitrogen cycle. And that's basically the premise of the talk today, is we're going to be looking at the interrelationship, the interactions between oxygen and nitrogen, especially at low oxygen levels. And the reason that becomes interesting is because the nitrogen cycle is very important in low oxygen environments, as you probably well know. And about a third of the fixed nitrogen that's formed happens in oxygen zones. And the question is, at least one question, would expanding ocean deoxygenation impact that production and the nitrogen economy of the oceans. So I'm not going to answer that question, but I hope I give us some tools by which we can try to look at it. So just to be different, I try another view of the extent of deoxygenation of the oceans. This is the oxygen concentration at the minimum values as we generally understand from the oxygen data that's available. We all know this and we've heard much about it, but I want you to pay attention to the numbers, because the slowest value we see on this slide is 5 micromolar, which is not very much oxygen, as you well know, but that doesn't really give us the picture. That really doesn't tell us about the oxygen levels, which are relevant for the environments we're talking about. And in order to give you some focus on that, some years ago, Niels Peter Resbeck from the University of Aarhus in Denmark developed techniques to be able to measure oxygen to extremely low levels, down into the low nanomolar levels. And when applying those techniques to oxygen distributions in the oxygen minimum zone, in this case off the coast of Peru, what you find is that oxygen levels aren't 2 micromolar, which is what the Winkler titration might tell you, or 5 micromolar as that map told you. It's actually less than 2 or 3 nanomolar in concentration. So that's really not very much oxygen at all. And this impacts our understanding about how oxygen is involved in biogeochemical cycling in these zones. And just to give some contrast, if we go a little bit further offshore off the coast of Peru, we start to see resolution indicating that oxygen levels are low here, but now they're in the sort of the 10 to 20 nanomolar range. So with this new technology, we're actually able to really understand the distribution of oxygen as it actually is in the environments. As we'll see towards the end, as this might not actually be good enough, but that's a conclusion for later. Now, Bo Tomdrup, my good colleague, had the opportunity to take a cruise up and down this eastern tropical South Pacific in sort of the hardy oxygen minimum zone and map out oxygen levels there. And instead of having oxygen distributions of 2 micromolar or 5 micromolar, you can see that the map is telling us oxygen levels are predominantly lower than 100 nanomolar in this region. So we're dealing with very, very low oxygen levels indeed. And now comes the hard part, and this you're just going to have to bear with me, because what becomes interesting when oxygen levels get low like this is we start to have the possibility for coupled aerobic and anaerobic metabolisms. We have the ability and the possibility for processes requiring oxygen, using oxygen to coexist with those that don't. And the processes that we're interested in are those of the nitrogen cycle, at least in this talk. And what I'd like to do is just explain to you a little bit about the structure of an oxygen minimum zone, at least as we see off the coast of Peru and Chile. And if we start here with oxygen, oxygen goes down to those low values that I've talked about. Nitrite typically accumulates to values which are insignificant, and they accumulate at the expense of nitrate. So you see a reduction in nitrate concentrations. Importantly, we often also see a double peak in chlorophyll A, occurs in the region where oxygen is basically undetectable. We're going to hear more about that from Amelia later on today. And it's predominantly, or actually not predominantly, it's a chlorophyll A maximum, which is dominated by cyanobacteria of the Prochlorococcus group. Okay, so that's that part. There's oxygen production even on these in these ostensibly anoxic areas at the top where the secondary chlorophyll maximum exists. And now we have a bunch of anaerobic processes that are also likely to be occurring in this zone. So we have the reduction of nitrate to nitrite. The anaerobic process doesn't require oxygen. This is a heterotrophic process, so we oxidize organic matter. In the process, we liberate ammonium. This is the ammonium that's part of the organic matter itself, of the organisms that are decomposing. The ammonium and the nitrite can combine in a process called anammox in order to produce N2 gas. So that's a loss of nitrogen from the environment. And we also have sort of standard good old-fashioned heterotrophic denitrification, where organic matter reacts with nitrate and nitrite in order to produce N2 gas. So we have these two pathways of N2 production, nitrogen loss. We also have aerobic pathways occurring in these zones. Potentially we do anyway. We have aerobic respiration, of course, standard aerobic respiration. We also have nitrification reactions of various types. So we have the oxidation of nitrite by oxygen back to nitrite by oxygen back to nitrate. And we also have the oxidation of ammonium with oxygen back to ultimately back to nitrate. And if you think of it, and this is just a little bit of an indication, sort of a warning of where we're going, is these processes of nitrogen loss, especially anammox, which it turns out to be the really important one, require that we have ammonium and nitrite in order to be able to operate. These processes remove it. So there's some sort of a competition in a way between these products, nitrate and ammonium with oxygen, and then the anammox process. And we're going to get into that in more detail later on. But before we do, what I want to try to do is just try to give you some indication about the oxygen limits for life. You know, you can ask this question: how much oxygen do heterotrophic organisms actually need in order to be able to function as viable organisms in the environment? And we set out to try to answer that some years ago by looking at an organism which was pretty well understood. It was E. coli, which may not be the most important organism in OMZs, but at least it's a start point. We grew E. coli. For those that know E. coli, it can do all kinds of stuff, but we grew E. coli with glycerol so that it was functioning solely as a heterotrophic aerobe. It couldn't ferment with this substrate. We put it in an incubation chamber, something like this, where we fed oxygen continuously through a silicone tube. And what we found was that E. coli would grow through this with a growth rate which depended on the rate which with oxygen was supplied through the silicone tube. And as it grew in the population size, oxygen was drawn down to concentrations of less than we could measure with the stock sensors. So at least in this particular case, we have aerobes that are able to grow with oxygen levels that are less than a couple of nanomolar. So you might ask the question, how low can organisms actually go? And from an ecological perspective, I want to present sort of a perspective that Emily Zakem had introduced. And I don't want to steal her thunder. She's talking a little bit later on today, and I'm sure she's going to talk about this and some other things as well. So I'm only going to brush over this lightly. But what she defined was something called a critical oxygen level. And that critical oxygen level, growth of an aerobic organism matches its loss rate by death, by viral attack, by grazing, by whatever process is removing it. And you can come up with a simple equation defining this critical oxygen level, and that includes understanding the loss rate of the organism. It understands you have to understand the volumetric carbon content of the organism, how much carbon's there. You have to know the size of the organism. You have to know the oxygen efficiency, how much carbon is produced into biomass during the process of respiration, the diffusion coefficient for oxygen. Okay, so you apply these. And we can apply those with the parameters that we understand from E. coli. And what we find is that here with this specific growth rate is the same as the grazing rate. It's the same as the loss rate. So with growth or loss rates, which are relatively slow, you can see that the organisms in principle can have critical oxygen levels in the sub-nanomolar range. So it's not unthinkable that we can have organisms living as aerobes in OMZs in environments where we can't measure oxygen, even within technologies that we have. And I would argue this, and I would add this, and Emily will probably get to this in her talk as well, these are maximum levels in a way, because this analysis doesn't include the possibility that an organism can be conducting both aerobic and anaerobic metabolisms at the same time. And if you think about that, an organism might be able to grow anaerobically, but still respire a little bit of oxygen. The oxygen levels lower than what this analysis would provide. So this is just to sort of put some kind of a perspective on it. Okay, now this is the oxygen part of the heterotrophic part of the oxygen story. And what I want to do now is into the nitrogen cycle. And with the nitrogen cycle, we analyze the oxygen sensitivity of this process a little differently. With E. coli, we're able to look at growth. That's really hard to do in sort of the experiments we've been conducting to look at the nitrogen cycle. So instead of growth, we're looking at metabolism. So the typical experiment is you take some sample from an OMZ that you're studying, let's say it's from the Eastern Tropical South Pacific, which is where we've worked, and you take that water and you subject it to different concentrations of oxygen, and then you look at the rate of the reaction. And if we do that with nitrifying, with the nitrification process where ammonium is being oxidized, you can see that you need to get down to rather low concentrations of oxygen before the oxygen concentration starts to impact the process. And this is a typical Michaelis-Menten type analysis where you find that the Km value, that value where the rate is half of its maximum rate, is submicromolar, let's say in the half micromolar range. The same is true for nitrite oxidation. So these processes can occur at very, very low oxygen levels at reasonably good rates. And not only that, we are able to measure these processes as occurring to oxygen levels as low as we can actually measure within our incubation chamber. So these processes actually can occur at very, very low oxygen levels. The flip side, now we have processes, the anaerobic processes of denitrification and Animox. And these processes, of course, ultimately might be inhibited by oxygen for various reasons associated with the enzymatic pathways that they conduct. So what we do is we look instead of looking at the oxygen regulation of the process in a positive way, we look at it in a negative way, and we describe oxygen inhibition. And what we find is that with the water that we incubated from the Eastern Tropical South Pacific, that denitrification becomes fully inhibited at oxygen levels of about a micromolar. Animox is much more oxygen tolerant. So the Animox process, the physiology of the Animox bacteria, they can tolerate much more oxygen than that. So if you want to keep something in your head, in something like the micromolar range, something less than a micromolar, Animox and denitrification be all right physiologically. You know, more than that, then it depends a little bit on circumstances. Okay. So that's sort of where we're at, and now what we want to do is to try to see if there's a natural environment we can go to try to understand how these processes might interact. Remember that the Animox process requires ammonium and nitrite, and ammonium and nitrite are oxidized by oxygen. So oxygen, even at low levels, impacts the Animox process in a way, but it also impacts the substrates that the Animox process requires. So it gets a little complicated. And to help us sort of visualize this, let's look at the Bay of Bengal. So we haven't talked about the Bay of Bengal yet in this conference. It's sort of an oddball as an oxygen minimum zone, because traditionally it's been recognized as anoxic because using Winkler titrations, you can't measure oxygen there. But it's also been recognized as an oddball because recognizable nitrogen metabolisms have not been found there either. So we went there with this new oxygen technology that Niels Peter Revsbeck developed, and so we were able to measure oxygen concentrations accurately at low levels with these so-called stock sensors, and those would be the blue dots here. And what we find is that the oxygen levels are persistently less than a micromolar in the Bay of Bengal, and in many cases sort of in the 10 to 100 nanomolar range. And this seems to be something that has been the state of the Bay of Bengal for many years, because active nitrogen, well, we'll get to that. This seems to be a long-term thing. I want to make a comment, and because this relates to oxygen sensing technology. You notice that we have this is the high sensitive oxygen measurements with the blue dots. We also have used seabird oxygen sensors, and that's the orange lines here. The seabird oxygen sensors are excellent. As you can see, they're able to distinguish nanomolar levels of oxygen. The problem with the seabird sensors is you can't calibrate them easily to zero. So we use the stock sensors to do that. But once you get them calibrated, out to be a very effective tool for looking at low oxygen levels, much lower than what they promise they can do. So just something to keep in mind practically, is these might have be very useful if we can figure out to calibrate them better. Okay, so back to the Bay of Bengal. So oxygen are low, but interestingly, we have microbial communities that are able to conduct denitrification based on gene abundance. They're able to conduct anammox process based on gene abundance, and we also have nitrifiers there as well. So we have the complement of organisms that can be doing the nitrogen cycle, but what are they doing? So we start off with the process of nitrate reduction to nitrite, because that's fuels, in a sense, the anammox process. We get nitrite that way, and we also get ammonium that way. Both those things required for anammox. And we see that at least in some of the sites that we looked at, 2 out of 3, there are reasonably rates of nitrate reduction to nitrite. These would be under environmental levels of oxygen. Okay. Now, what about the process rates? What about anammox and denitrification? One, we didn't measure any denitrification anywhere. So we tried and we couldn't. Anammox, black dots. Black dots are the anammox rates that we tried to measure under in situ conditions, and we used the ammonium. In this case, you can use labeled ammonium or nitrite to measure anammox. We used ammonium. No anammox, couldn't measure anything. But when you add nitrite to the system, we always then were able to measure some anammox. So what this tells us is that anammox was nitrite in the Bay of Bengal, even though nitrite was being actively produced by nitrate reduction to nitrite. The conclusion there would be that these oxygen levels that we have in the Bay of Bengal, these oxygen levels of, let's say, 10 to 100 or 10 to half 500 nanomolar, are sufficient to actively oxidize or actively support nitrification that oxidizes the nitrate completely from the water column and makes it uncompetitive for the anammox bacteria. So even those small levels of oxygen are sufficient to remove nitrite from the anammox bacteria, even though, and this is important, the anammox bacteria themselves are not physiologically challenged by those levels of oxygen. They're fine with those levels of oxygen. They don't care about that, but remove a key substrate, even those low levels of oxygen. Okay. Now, I'm going to get to something a little bit complicated, and I hope this is okay. But And I have to take you through the stepwise because it's a tiny bit complicated. What I want to do in this analysis then is to figure out basically what is controlling the amount of anammox into production in these systems. And we start with the carbon cycle. And in order to constrain the carbon cycle, what I've done is I've looked at the export production of organic matter as measured through sediment traps. That's not our work, but that's work that others have done. I apply, for those that are familiar with it, an attenuation of the carbon degradation rate with depth using a Martin curve or something like that, if you're familiar with that. If you're not, it doesn't matter. It's just that organic matter gets utilized and consumed as it settles in the water column, and you have less organic matter after you go a certain depth in the water column, and these things are characterizable. These lines represent the amount of carbon oxidation that you expect in this part of the OMZ, given those parameters. Okay, now Andrew Babin, and also Loreto Bambrera from our group have done some excellent measurements of anammox in these environments. So anammox needs nitrite and it needs ammonium. Well, there's tons of nitrite here. That's there. Ammonium. Okay, so ammonium comes from the oxidation of organic matter. So what we can do is we can sort of convert these anammox rates into potential carbon oxidation rates. We say, okay, we need this carbon oxidation in order to support the anammox rates that we measure. Okay, does that make sense? Okay, we do that dots here, and what we see is when we're below 100 meters depth, that those rates act. The amount of animox that we measure in these rates is I think, clearly supported by the rates of carbon mineralization. In the carbon mineralization is most likely nitrate reduction to nitrite. And Andrew Babin, again, has measured that in a few cases. And you can see that at least below 100 meters, these are the black dots, those rates parallel very nicely with both the animox rates and our estimates of carbon mineralization. Up here, they don't. The rates of nitrate reduction and nitrite are low. And one of the reasons might be true is because in this level here, when we're up at the top of the oxygen minimum zone, we have this active production of oxygen by these deep Pleurococcus communities. They might be competing with aerobes, right? These aerobes that can be in principle operating at very low oxygen levels. So there might be between different clades of organisms, or organisms using different both aerobic and anaerobic strategies. Anyway, the point is when we get above 100 meters, you can see that these green dots don't follow these projections, these estimated carbon oxidation rates at all. What that means is we're missing animox. There's animox that's missing there. Potential animox that we don't see, that we don't measure. I think that this is true is because even at these very low measure, it seems logical, at least possible, that these oxygen levels are oxidizing the ammonium that's being produced through carbon mineralization. So we have an active process of sort of cryptic. We don't really see it. That's removing the potential for animox here, and I think global change context or a future change context, we're talking about basically half of the potential animox which we could have operating, at least at this particular place. And understanding how this dynamic works might be really critical for us to be able to predict how this dynamic might change or might not change in the future. So I think this is a real and interesting science opportunity, is to try to figure out what's going on here. I think that's really, really cool. So if I try to summarize what I've been talking about so far, I try to do it with this graph here, where I have these different processes, the anaerobic and the aerobic processes, and the color kind of indicates the magnitude of these processes as a function of oxygen concentration. So let's start with the anaerobic processes. They are not really bothered until we start to get at oxygen levels above 100 nanomolar, or maybe even a micromolar. So as long as we're in this range here, these processes should be physiologically possible. Okay, now what about the processes of nitrification? You know, where we're removing ammonium and nitrite. Well, when we have high oxygen levels, they're good. When we get oxygen levels like we have in the Bay of Bengal. So this is why we don't have animox in the Bay of Bengal, because the ammonium and nitrite are being removed by nitrogen, even though the bacteria that conduct animox and reduction and denitrification are fine, but the substrates aren't available. When we get to the top of the OMZ, that place we were just at before, of course, the anaerobic processes are in good shape, but we still seem to find some removal of the substrates available for them. And I'm going to argue that it's oxygen related, and the oxygen levels are probably in the range of an nanomolar or less, levels below where we can measure now. And then when we get into the 4, then we seem to be free of oxygen production, and then the processes can occur as they should. But life is getting very complicated and very interesting. I'd like to highlight just one more thing before I'm finished. Meet Nitrosopomelos maritimus. This is a nitrifying organism. It's one of the most abundant organisms in the ocean. It's a taumarchiota. It's very well understood in many ways, its physiology. And what it does is it takes ammonium, combines that with oxygen, produces nitrite, and it does this all over the global ocean. Well, yeah, maybe. There are certain parts of the global ocean, like the Black Sea, for example, that's where Nitrosopomelos, which is the pink dots here, are found in a region which I'm not going to call suboxic. I'm going to call it anoxic, nitrogenous, or anoxic non-sulfidic. I hate that term suboxic, and there's some people here that know that. Anyway, that's an aside. What you find, you have a system here in the Black Sea where you don't have any oxygen, but you have these Tamarchiota in high abundance. Well, it doesn't seem possible that they're conducting their typical aerobic processes here. They must be doing something else. So now enter Beate Kraft, who is a postdoc and now a colleague of mine at the University of Southern Denmark, and she was playing around with nitrous pummelose because she was interested in trying to figure out its low oxygen. How did that, like some of the other experiments? But she found that it did this curious thing. When she was doing experiments where it was drawing down oxygen to very low, it would bring oxygen down to very low levels, 0 basically, and then it would start to accumulate again. And that was a very repeatable observation. It's been repeatable over 5 years observation. So it's something that's very characteristic of how this organism metabolizes. If we add cyanide, which is a blocker for the oxidase enzyme, then oxygen increases quite substantially. So nitrous pummelose is producing oxygen. You can read our paper if you want to go into more details about that. We published it recently. We checked everything. You know, we spent 3 years or 2 years just trying to figure out if this was contamination or was add some other way that we could be producing oxygen in the system that wasn't related to the organism, and we convinced ourselves that that wasn't true. So we have dark oxygen production by nitrous pummelose. This isn't the first time, though, that dark oxygen production has been observed in nature. So there's this fairly well-known now, the NC10 bacteria, I always stumble over this name, Methylmeribulus oxifera, and it's a real interesting organism because what it does. It's a methane oxidizing organism, and what it does is it converts nitrite to NO. It dismutates the NO to oxygen and N2, and then it uses the NO to oxidize methane. This organism isn't known to allow oxygen to leak from its cells. All this is internalized within of the organism, but it's another example of this oxygen production. So we were curious as to whether our organism, nitrous pummelose, was doing the same thing. So we found out that it wasn't quite exactly. So this is an experiment where we looked at N2 production, and we monitor also oxygen accumulation in the system. And if it was doing the same thing as the NC10 bacteria would do, we would see oxygen production and nitrogen production at the same time if it was all coming from the dismutation of NO. But what we saw was that oxygen production was occurring before N2 accumulated. So something else was going on. We found out that N2O was accumulating, and just to make a story, we decided, or at least decided, we hypothesized that the organism has a metabolism like this in the absence of oxygen, where it's taking nitrite, it's getting electrons from someplace, maybe organic matter, it's nitrite to NO, the NO is dismutated to oxygen and N2O, and then further converted to N2. And the reason that it's doing this is because it still wants to be an ammonium oxidizing nitrifier. So the oxygen that it produces, at least most of it, is cycled back into oxidizing ammonium. We also showed that. But the cool thing about nitrous pummelose is also it leaks some into the environment. And I think that there are a lot of really interesting questions that arise, and that is, could this help to fuel aerobic metabolisms in the heart of oxygen minimum zones? Are there other organisms the same thing? Maybe this is a more common type of metabolism that we think of. There might be a dark world of oxygen production down there, which is something which is new and exciting, but we just don't understand very well. So I think that with that, I'll acknowledge many people who have helped to produce the data, so the ideas that I've presented here, and I thank you for your attention.
right, thanks, Don, for a very interesting talk. Have time for a couple questions.
Excellent. Thanks, Don. You haven't mentioned sulfur other than in passing. Do you think it could play a role in the Bay of Bengal, Arabian? You started off with the Bay of Bengal and no denazification. Is sulfur playing any role in that?
That's a really interesting question, Andreas. We didn't check that. So I can't say for sure, but my sense is that the oxygen levels are high enough there that any oxygen, sulfur metabolism would have difficulties unless it was associated with sinking particles. And there it would be possible. So that could be a possibility if it was confined to micro niches and particles. Yeah. We didn't look for that.
Carolyn?
Yeah, thanks for a great talk. I had a question about the Bay of Bengal, where you showed that there are microbes able to do anammox, right, in the water column. So why are they there if they're not doing anything?
That's a really great question, Carolyn. I think that the answer must be that in time or space, anammox occurs in the Bay of Bengal. And it could be that there are parts of the bay where there are somewhat less oxygen than what the water gets transported in, or just on some time scale, those last little bits of oxygen. So, I'm glad you mentioned this because this actually brings up a point that I forgot to say. The Bay of Bengal is on a tipping point, right? We have these very, very low but measurable concentrations of oxygen. Those last half a micromolar of oxygen could be removed. We've calculated that the Bay of Bengal could be as important as the Arabian Sea in nitrogen production. So it's really important that I mean, who knows what's going to happen in the future, but there's something, there's a system right at the tipping point.
Okay, thanks.
I think we have one online question from Aurélien. I don't know if you can activate your microphone and ask your question. Otherwise, I will read it.
Yes. Okay. Thank you for your very nice presentation. Yes, I have a question. What about the growth rates of the different aerobic and anaerobic communities you mentioned?
I'm sorry, could you repeat that again?
What about the growth rates of the different?
Yeah, so that's a question. I think with respect to the processes like anammox and denitrification, we are probably measuring growth rates. At least we think we are in those processes. With respect to the rates of carbon mineralization that I've estimated, based on sediment trap work, those aren't growth rates or anything, but just estimates based on ideas on how carbon should be cycled. But I think that we're measuring growth rates when we're measuring actually rates in the environment. Although, I mean, there could be some recycling going on there as well. But
okay, thanks.
Okay, well, let's thank Don again, and if you have more questions for him, maybe catch up with him at the podium. All right, so our next speaker is Yu Han, who is joining us from Xiamen University. Yu, I think you're muted. We can't hear you. I think you're still muted.
Hi, everyone. Can you hear my voice?
We can hear you now.
Okay. let's start it. Hello, everyone. It's my great honor to attend this conference. It's my pleasure to share my work online with so many famous scientists today. And the title of my study is Transcriptomic Evidence for Microbial Carbon and Nitrogen Cycles in the Deoxygenated Seawaters of Bohai Sea. A statistic indicates that dissolved oxygen levels in the ocean has been persistently falling over the past 50 years. The studies estimate that the global ocean is expected to lose 3 to 4% of oxygen capacity by this century. Subsequently, hypoxia has inevitability occurrent in some areas. Hypoxia is generally considered to be dissolved oxygen concentrations less than 2 milligrams per liter. Dissolved oxygen reduction will directly threaten the growth and the survival of aerobic organisms in the aquatic ecosystems. Also, the decline of dissolved oxygen will also constrain ecosystems energetic, alter the ecosystem structures, and drastically affect microbial mediant biogeochemical circling of elements such as the nitrate reduction and sulfur oxidation. Climate warming is one of the non-ocean dioxygenation mechanisms directly describes oxygen solubility slows down the water circulation while intercepted stratification and also promote the microbial respiration, resulting a decrease of dissolved oxygen. Excessive nutrient discharge induced eutrophication in coastal waters will promote microbials representation in both water column and sediments, consuming a large amount of dissolved oxygen loading to the coastal ecosystem facing more serious deoxygenation environmental problems. Scientists indicate that more than 700 coastal zones has been experienced hypoxia. Microorganisms, including bacteria and archaea, are highly abundant with an extremely large collective biomass. Ocean deoxygenation will force ecosystem material and energy flow to shift from higher level predations towards to lower level microorganisms. Studies has indicated that microorganisms can still grow aerobically in low oxygen environments and even under hypoxia concentration. However, the adaptive mechanisms and the environmental regulating factors for microorganisms in the oxygenated waters has regularly been investigated. Well, as we know of these three methods have generally been applied to study the microbial community and their related functions. Well, most studies for the correction of archaea is limited due to the primer design. Then our study map selected as a typical coastal area of China has experienced seasonal deoxygenation events with low oxygen and hypoxia zone, generally observed from June to September. In my study, we select the samples through filtration and investigated the dynamics of community composition and transcriptional activity of dominant bacteria and archaea in Bohai Sea. Therein, we found the dissolved oxygen decreased to a value of 3.2 milligrams per liter in bottom water at seat A45. Well, about the environmental condition, dissolved oxygen, pH, and temperature presented a decreasing trend with depth. The combination of declining ammonium concentration and rising nitrate concentration with the water depth. Apart from that, the totally prokaryotic abundance also decreased significantly with increasing depth. About the microbial composition was showed 3918 OTUs that belong to 45 phyla. The dominant group were proteobacteria, Firmicutes, and cyanobacteria. At the family level, Firmicutes was belong to the coastal ecosystems, natural supermulaceae. Cyanobacteria were belong to the FAM2 group, and the heterotrophic bacteria were mainly distributed in the families such as Polygibacteriaceae, Rhodobacteriaceae. And the significant difference analyzed found at a variety of taxonomic level with the water depth increase and the dissolved oxygen decreased. Firmicutes, especially the family Supermulaceae, the abundance and the transcriptional activities significantly increased. Instead, that of cyanobacteria significantly decreased. So what the environmental factors are responsible? The results showed that, the results showed that dissolved oxygen, pH, temperature played an essential role in regulating the composition and their activity of microbes. These three factors were negatively correlated with Firmicutes, but positively with cyanobacteria. Oh, that's going to the functional diversity of bacterial community. There will be some delay. We got 2.2 billion from 33 samples. Specifically, the core coding genes of the ammonia oxidation progress was significantly increased. Correspondingly, the enhanced transcripts in ammonia transporter and ammonia assimilation. This indicates an increased demand for ammonia by Firmicutes. On the other hand, 3HP to 33 folds in the low oxygen water. This carbon fixation pathway is far more energy efficient than the CBB circle. It suggests that canotrophic carbon fixation pathway will be strengthened in low oxygen environment. Nevertheless, a significant decrease in cyanobacterial abundance and transcription of gene related to photosynthesis and carbon fixation pathway. The pattern of carbon fixation shifted from photoautotrophy to canoautotrophy in the low oxygen. And a large amount of transporter coding genes was also found here. The transport various nitrogen-rich compounds such as ammonia acids, peptides, nucleosides, and vitamins, while carbohydrate and high molecular weight lactobacillus were relatively less. It was found that there were abundant dissolved organic nitrogen transporters in the hypoxia zone, and there may be ammonia mineralized from dissolved organic nitrogen to maintain nutrient circle. And we also focus on antioxidant coding genes from major microbial groups with decreasing concentration of dissolved organic, the oxygen, the abundance of antioxidant transcripts, including superoxide dismutase and catalase in heterotrophic bacteria and cyanobacteria. In the bottom water is lower than that. However, the transcripts of superoxide dismutase and low efficiency protein scavenging coding genes proceed redundant. In the 5-methiota increased by 13-fold in low oxygen waters regardless of life cells. Then we did the normalization analysis showed that with the decreased, with the decreased oxygen, most of the transcripts of the antioxidant genes increased, responding to the increased transcript of electron transport chain and ammonia oxidation progress. It suggests that the interest of intercellular reactive oxygen species probably stimulated by active metabolism under the low oxygen environment, and the enhanced antioxidant transcripts will elevate their adaptation. Finally, in general, our finding indicates that 5-methiota, especially the coastal ecotypes capable of ammonia oxidation, have great resilience to the low oxygen aquatic environment, and their chemically autotrophic carbon fixation contributes greatly to the carbon flow under the deoxygenated environment condition. And this work is part of my doctoral research. I would like to thank Dr. Zhao and Dr. Tang, my PhD supervisor, and also thank to the National Key Research and Development Grant Program of China and the National Natural Science Foundation of China for supporting my sampling. That's all. Thanks for your attention.
Right, thanks you for a great talk. We have time for one question, I think. Yeah, Lisa.
Thank you for that interesting talk. Do you know what sort of stable isotope, carbon stable isotope signature that carbon fixation by the Thaumarchiota would produce if we wanted to track it in the system and into animal life?
Oh, it's an interesting question. Well, because I did most research is about the microorganism. Actually, didn't consider too much about the animals in the seawater. Can you know, this maybe I didn't thought before.
Okay, thanks a lot. Let's thank you again.
Okay, thank you. Thank you very much.
Okay, our next speaker is Julia Huggins from the University of British Columbia.
Great, thank you so much. Pleasure to be here today. I appreciate being invited to this session. My name is Julia Huggins. I'm a PhD student with Sean Crow. I'd like to acknowledge all of the sources of funding and folks that have made this research possible, including the fieldwork, an amazing group of people who've come out in the ocean with me a bunch of times, and some folks that have helped me with lab work. I also want to acknowledge that all of the lab and field work done to make this research possible is conducted on the unceded territory of Canada's First Nations peoples. Okay, so as oxygen declines in environments, energy transduction is shifted to microbial metabolisms. And some that oxygen decline, microorganisms eventually shift over to using alternative terminal electron acceptors. In marine environments, that's nitrate and nitrite, as Don's talk very helpfully set up for this. And this transition from aerobic to anaerobic metabolisms, again, as Don very helpfully explained, a bit more complicated than our simple models would suggest. And that to competing pathways and some transition boundaries that are a bit unclear. But if we want to improve our models that help us qualitatively and quantitatively predict what happens in these marine environments as oxygen declines, we really need to improve our constraints on microbial metabolic activity under these low oxygen conditions. So that's where my talk is going to go today. So a major point of complexity here in this transition is that there's actually 3 major pathways of nitrate and nitrite reduction. As you can see here in my simplified version of the nitrogen cycle, anammox and dissimilarity nitrite reduction to ammonium, DNRA, are all possible paths of nitrate reduction. These 3 pathways have different influences on their environment. And it's important when we're predicting the outcome of deoxygenation to know which pathway or pathways becomes active as oxygen is depleted. One of the most notable differences between these 3 pathways is their influence on the fate of nitrogen in the environment. So typically when we're talking about oxygen loss in marine environments, we focus on denitrification and anammox, which are nitrogen loss processes because they produce N2 gas, which is not biologically available. DNRA, however, retains nitrogen in the environment because it bypasses N2 gas and produces ammonium. And our quantitative models actually show that this has a positive feedback on primary productivity, which can then further exacerbate oxygen drawdown and push the system toward sulfitic conditions. Unfortunately, DNRA is also the least well studied and understood process of these 3 different nitrite reduction pathways. And so our goal here is to improve our knowledge about the role of DNRA in low oxygen nitrogen cycling. So when studying a microbial metabolism, we can investigate the process at multiple stages across the central dogma of biology. We can use process rate quantify the actual microbial activity in an environment. We can also use metatranscriptomics to tell us something about the functional genes, specifically those related to the processes we're interested in and whether those genes are being expressed. And we can also use metagenomics to learn more about the organisms in that environment doing the processes we're interested in. So our work here, we actually use a combination of all three of these approaches because they each tell us different and valuable pieces of information. We conducted our research in Saanich Inlet. This is a fjord on the southern end of Vancouver Island on the west coast of Canada, in British Columbia. Saanich Inlet has a deep basin, which you can see remains anoxic or very low oxygen throughout most of the year, although it does experience periodic events of low amounts of oxygen introduction into part of the water column. Saanich Inlet is close to our university. It's very well studied, and it has a fairly predictable seasonal pattern, which makes this environment a very useful place for us to study oxygen deoxygenation and low oxygen microbial metabolisms more broadly. So as part of a larger time series experiment, we conducted a series of monthly cruises throughout 2015 and 2016, where we measured the geochemistry of the Saanich Inlet water column, nitrogen cycle process rates, and omics information about the microbial community in Saanich Inlet. So for a little bit of context here, this is what the oxygen concentration distribution looked like over depth and time for the period where we did these experiments. As we expected, we see high concentration of oxygen in the upper portion of the water column and nearly complete or complete drawdown of oxygen in the lower part of the basin, with a few low oxygen introduction events, both in the fall of 2015 and again in the fall of 2016. Nitrate concentrations, the distribution followed a similar pattern to oxygen, and you can see the oxygen transition zone marked here with red contour lines. And so nitrate concentrations, again, followed a similar pattern, high availability in the upper water column, and nearly complete or complete drawdown in the lower part of the water column, although we do see a little bit of nitrogen accumulation after the oxygen introduction event in 2016. And so over the same time period, we conducted a series of isotope labeling experiments to measure the process rates of multiple pathways of nitrate and nitrite reduction, namely the three I talked about: denitrification, Animox, and DNRA. So we did measure denitrification and Animox. I'm not going to show them here because the story is about DNRA, but we did measure those as well. So what we see for DNRA is that this process is definitely active throughout the lower portion of the water column in Saanich Inlet and across different times throughout the entire time that we were doing these experiments. I will point out that these rates are extremely high. They are higher than anything reported in a marine environment previously. And our highest rates of DNRA were nearly an order of magnitude higher than our highest rates of denitrification. So even though there's quite a bit of seasonal variability in rates, they're sufficient that when you integrate over depth and time, DNRA is the dominant pathway of nitrate reduction, at least in this environment. I also just want to call your attention to the fact that some of the highest rates of DNRA that we observed were in waters with low but appreciable oxygen concentrations. And again, thankfully, I appreciate the useful setup from Don's talk. As we know, there could be co-occurring aerobic processes such as nitrification happening in the same environment as this DNRA. All right, so we wanted to know a little bit more about the organisms that are responsible for this process. And so we turned to the omics, the DNA and the RNA that we extracted from select depths at these two different time points. So we looked at some samples from the spring of 2016 when the water column was highly stratified, as well as the fall leading up to and during that oxygen introduction event in August, September, and October of 2016. And the first thing we want wanted to do was to also to corroborate these rates that we observed. We wanted to look for the functional gene that I'm responsible for DNRA. And that's NRFA is the gene, the functional gene here. And what we see is that, yes, indeed, NRFA is both present and actively expressed throughout most of the water column, throughout most of the year. And we see that the waters where we observed high rates of DNRA have the expressed metabolic potential for this process. And interestingly, we see that there's also the expressed metabolic potential in waters where we didn't observe DNRA. And this could be due to a number of different factors that we can't fully constrain at this point, including facultative metabolisms, constitutive expression of genes, or even potentially cryptic cycling with co-occurring processes like anammox or nitrification. So gene abundance alone, however, doesn't actually tell us about the organisms themselves. And what we did next was we looked at these NRFA sequences, the sequences for the genes NRFA, and we manually curated them to identify unique phylotypes, unique versions of this gene. We found 20 different phylotypes of NRFA. And so what you're seeing here is, again, NRFA gene copy number abundance, both in the metagenome and the metatranscriptome, but this time split up by a phylotype diversity of those genes. And as you can see, a few phylotypes dominate both the metagenome and the metatranscriptome throughout both depths and over time. And notably, phylotypes Y and E showed up in all of our metagenomes and metatranscriptomes, often at a high proportion of the community. A few of the other important phylotypes I've highlighted here as well. We were not able to, just based on the phylotype alone, assign taxonomic information or taxonomic identities to these organisms. So what we did next was we reconstructed population genomes using the community sequence information in the metagenome. And so we do this with a combination of bioinformatic binning tools, and we're able to get a pretty good look at the organisms that were present in our environment in these population genomes. And then we look through those population genomes to try to find ones that contain the phylotypes of NRFA of interest in our system. And we were actually able to identify organisms hosting most of our main dominant versions of NRFA. And I just want to point out the two that I had talked about before. So the phylotype E NRFA is hosted by an organism in the Marini microbia group. And we know that the Marini microbia are metabolically diverse organisms that are ubiquitously associated with low oxygen OMZ environments globally. And then phylotype Y, which was our most dominant and actively expressed version of NRFA, hosted an organism in the Lentomicrobiaceae group, which is a relatively newly described lineage and not very well described or characterized yet. We do know, however, that the broader group that they belong to, the Bacteroidota, are widely associated with low oxygen environments, generally speaking. In addition to this taxonomic information, we can also use these population genomes to learn more about the other metabolic capacity of these organisms. And so, and we want to do that because it can tell us more about what regulates this process DNRA on a broader level, if we can understand what else this organism can do and what it needs and what its role in the environment might be. And so just as an example, I'm going to show you what we've learned from the population genome for the organism hosting phylotype Y in the Lentomicrobiaceae group. So what you're seeing here is a schematic of a cell. We've got the inner membrane and the outer membrane, and then the internal cytoplasm. And on this are highlights from the metabolic capacity of this organism that we know from looking at the population genome. And so what I want to call your attention to is that, oops, that is not the right button, is that in addition to the capacity to reduce nitrate and to conduct the DNRA process. These organisms also have the capacity to use oxygen as a terminal electron acceptor and to respire aerobically. And notably with a low affinity terminal oxygenase version of cytochrome C. And they also have the capacity to metabolize a broad range carbon substrate through, am I back on? Through the citric acid cycle, including probably starches, sugars, volatile fatty acids, acetate, and ethanol. And in addition to that, they can use sulfide as an electron donor. So what this tells us is that this organism and the DNRA process that it carries out is probably regulated by electron donor availability, such as carbon sources or potentially hydrogen sulfide, as well as the potential aerobic activity of this organism, especially if it exists in that low oxygen transition zone with the capacity to switch back and forth between oxygen and nitrate as an electron acceptor. And in addition to that, this may implicate the Lentimicrobiaceae group as an organism involved in the sulfur cycle, for those of you who are interested in that. All right, so in conclusion, we see that DNRA may be a major mode of microbial respiration in low oxygen marine environments. And this challenges some of our major assumptions about the feedback from deoxygenation. Notably, for those of you who might go looking for this process, even when you design your experiments and are willing to carry out the extra more complicated steps to measure DNRA, it still might be overlooked due to high levels of seasonal and/or cryptic cycling with co-occurring processes. So keep that in mind as you're designing your experiments. And then lastly, DNRA is supported, at least by some organisms, that are facultatively aerobic with diverse metabolic capacity. And then lastly, just saying that going forward, sort of the last piece of this puzzle for us is to try to extend or assess the global, looking at the distribution of these NRFA hosting organisms in other low oxygen marine environments. Thank you very much.
Okay, well, we'll just go. Oh, there we go. All right. So yeah, thanks for having me. I was actually at the Liège colloquium in 2014, so it's exciting to be back. I wish I could be there in person. So I like this image here. It shows oxygen at depth in a model because it highlights the small scale circulation as being super important for the formation of these low oxygen zones, as well as the equatorial jets. All right, so we know that, oh, I did the thing that I wasn't supposed to do. Okay, so we know it's a combination of both biological and physical processes that form anoxic zones. So sinking of organic matter provides the fuel, and then low rates of oxygen supply laterally result in the depletion of oxygen. Characteristic anoxic zone profiles, so where oxygen is depleted, nitrate has a chunk out of it because it's used as a terminal electron acceptor, nitrite accumulates, and then we have this secondary, which Emilio just talked about, where we have evidence that there's cryptic oxygen cycling, right? Oxygen is produced and consumed there, and there's presumably anaerobic processes happening there too. So today I want to ask the question for this coexistence of aerobic and anaerobic activity at places like the SCM, why they're competing for electron donor substrate. How could it be that both exist at the same time? Does it require vertical mixing through the oxycline? And I'm also going to ask, why is there an SCM in the first place? Okay, so how do we answer these questions? So ecosystem models are useful tools. They help us organize, quantify, synthesize, and untangle complex interconnected processes and feedbacks. So there's different ways to use models. I feel like it's important to differentiate these two ways. It's not a dichotomy, but one way of thinking about models is what I'm calling a linear use, where there's observations, experiments that gather the data into a model, and then the model is used to make predictions. And that's important use of models. But what I'm doing today is different. I'm using models in this iterative way, and it's data from observations and experiments. And both in this way, both models and data contribute to theory and a better understanding of the system. So I'm coming from an overall strategy of building towards first principles of microbial ecosystem. So you think of a cell as being fueled by substrates that are taken up into the cell. Some of that is channeled into biomass synthesis or growth, and a lot of that is respired and excreted in modified form, which is the biogeochemical cycles that we care about. So in principle, we can use physical constraints to describe the uptake of substrates into a cell. And the chemical is the redox reactions and electron flows to partition the amount of substrate going into synthesis or anabolic versus catabolic processes. So back to the question, what allows for aerobic and anaerobic coexistence? Again, we said it's this relative ratio of organic matter versus oxygen supply into these zones that controls the formation of them. So the simplest way to describe that system is with a virtual chemostat, where we have a vat, and we can control the rates of oxygen and organic matter flowing into the system. So we can seed then that chemostat with different microbial players. And for right now, I'm going to show you an outcome of a simulation where we have an anaerobic and aerobic heterotroph competing for organic matter. And we could do this with any competing pair, competing for an electron donor, such as nitrite. I mean, Amelia just talked about how there could be aerobic and anaerobic nitrite oxidation, but here we're just thinking about organic matter as an electron donor. These model simulations are equivalent if I model a facultatively anaerobic population where the population switches back and forth between the use of oxygen and nitrate. The results are different if it's possible for the facultative population to do both at the very same time and use less oxygen. But we can get into that later in the questions if you want. But for now, it's equivalent if we think about facultative populations. So we're going to model the growth and the losses of these metabolic functional types, where the growth is dependent on a mechanistically modeled rate of uptake of required substrates into the cell, as well as an estimate of the yield or the efficiency of the use of each of those substrates towards channeling towards biosynthesis versus respiration. And so this allows us to look at solutions that don't rely on prescribed oxygen thresholds or critical oxygen concentrations. Rather, we can look at the solutions that come about due to the ecological interactions, these types with each other and with their environment. So these are the solutions at steady state. So on the x-axis, it's not time, it's not space, it's different ratios of oxygen versus organic matter that are input into the chemostat. And then I let that run and I look at the steady state outcome that results and plot that. So these lines are composed of many different dots of different steady state solutions. So we can see there's two regimes. So we're tying this now to this longstanding ecological theory, resource ratio theory that's developed, at least for natural environments, by David Tillman back in the early 80s. So on the left side of this dashed line, there's a domain of competitive exclusion by the aerobic population. Plenty of oxygen, it accumulates because there's extra, so to speak. And the aerobic population has competitively excluded the anaerobic population, or metabolism for the facultative case. And this is because we assume that because of their underlying reactions, that anaerobic metabolism is slightly less efficient at metabolizing organic matter into biomass. And this just describes most of the ocean as we know it, right? We know that and metabolisms dominate when there's oxygen. And then past a critical threshold here, which is set by the ratio of incoming oxygen to organic matter or electron donor, then we enter a domain of stable coexistence. So this theory and the way that this mechanistic model plays out is that there's not exclusion of the aerobic metabolism, but rather both aerobic and anaerobic metabolism coexist stably, with the ratios of aerobic to anaerobic activity being driven by the oxygen to organic matter supply. And all this happens, this coexistence happens while oxygen is down at very low concentrations. So here, this O2 star line, this dashed blue line, is the resource subsistence concentration of the aerobic population, which is what produced in the first talk, which you can understand as a function of the traits of the organism, including the parameters that inform the uptake of oxygen into a spherical cell. And so in these seemingly anoxic conditions, you have potentially very high rates of aerobic activity and varying rates of anaerobic activity. This might be on the sort of right past the dashed line. This could be where the secondary chlorophyll max is, right? That could describe that situation rather well. And then more to the right of here, where there's very low, but maybe intermediate, every now and then there's like a poof of oxygen that's supplied. That could be more like the core of the anoxic zone, where there's nearly exclusively anaerobic activity. Okay, so that's the stable coexistence. So it says that we don't actually need mixing of different of oxygen or different biopopulations in order to see both aerobic and anaerobic competing metabolisms. So next, I'm going to show you what happens if we expand the metabolic resolution of this model and resolve 6 of the nitrogen cycling metabolisms in or near anoxic zones. So we're going to still have our aerobic heterotroph type. By the way, these are meant to aggregate across many different species, right? Like in climate models that resolve just 2 or 3 different types of phytoplankton. Here, just like we know that there's a whole community with, KM values, et cetera. But here we're just trying to get the essence of what happens for at a broader level by assuming that all of these are aggregated into single types. So we have a nitrate reducing heterotroph that produces NO2, a denitrifying heterotroph that takes NO2 and converts it into N2 or N2O, and then 3 chemoautotrophs, ammonia and nitrite aerobic chemoautotrophs, ammonia and nitrite oxidizers, and an anaerobic ammonia oxidizer. It's going to be really interesting to expand the system to include anaerobic nitrite oxidation. I think we could do that pretty easily. So how do we parameterize? This is where that redox chemistry comes in useful. We can use observations and the underlying key half reactions of the redox reactions that underlie each of these metabolisms to put together these metabolic budgets that relate the amount of substrate that's taken up by each metabolic functional type into biomass, and then also relate the amount of excreted waste product. So this redox chemistry is really useful. This allows us to quantify and the relationship between biomass and substrate flow, which is essential for if we're going to ultimately link this to gene abundances or other sequencing data sets, right? So here I'm showing you budgets that are all normalized to 1 mole of biomass formation in terms of nitrogen. And you can there's a lot to take in, but I just want to point out just some key differences. So the chemoautotrophs, the aerobic chemoautotrophs require a lot more oxygen than the heterotrophic aerobe, which is how we understand that. And then we also noticed that the heterotrophic types all produce ammonium. And so these are all stoichiometrically and electron balanced. So now we have a framework that ties together this complex nitrogen cycle, resolves the excretion of ammonium, resolves the relationship for anomox between the amount of ammonium and nitrite taken up, for example, in ways that we can compare against observations, and we can show that these make sense. Okay, so those are the yields that inform the relationship between substrate and biomass formation. We can also increase the resolution of the physical environment. And here, and we know the circulation is much more complicated than this, but as a cartoon, we can capture the essence of what forms an anoxic zone with a 2D circulation model. So we see this just was this passive transport of a tracer, that there's this intensified equatorial upwelling, Ekman transport, and dispersed downwelling. And this mimic combine the circulation field with an ecosystem model where we have our 6 types as well as phytoplankton types, zooplankton grazers, sinking particulate organic matter, 3 species of DIN and oxygen. And again, we could run this to equilibrium state where we don't have any critical oxygen concentrations. Rather, that's the evolution due to the ecological interactions between all of these types and players. And we find that we are able to simulate the oxygen profile rather well qualitatively. And here, there's a lot to talk about with this model, but I'm just going to show you the biomass concentrations that result of those 6 key nitrogen cycling players. So everything is where we think it is at ratios that are somewhat realistic. So we have the 3 aerobic types that are all populating the oxygenated portion of the regime, and they've chewed down the oxygen and created the habitat for the anaerobic types to persist. The ratio of denitrification to anammox is, well, sorry, anammox to denitrification is something like 30%, which is other theoretical and observational papers have shown that that's the case. And yeah, like I said, there's a lot more questions that we could ask of this model, but right now what I want to just point out is I want to take a profile through this anoxic zone, which is here. And you can see that there's the formation of the secondary chlorophyll maximum. Now I've jumped to the head a little bit because I haven't told you how I've allowed this to be simulated, but I'm going to tell you this now. So I've used this model framework to explore a hypothesis for what forms the secondary chlorophyll maximum. In the simplest understanding of any system of mechanistic description of how biomass forms in the environment and is sustained, we can understand the resulting phytoplankton biomass as being a function of the rate of nutrient supply, as well as an inverse function of its loss rate or losses to grazing, virolysis, other mortalities. So here, if chlorophyll concentration, if the phytoplankton biomass follows the same pattern as the chlorophyll concentration, which it might not, but if it does, there needs to be some way that this phytoplankton biomass increases in the anoxic zone. How could that be? The only way that I could think of, because nutrient supply shouldn't increase, right, across that gradient, phytoplankton are there the whole time to consume it, would be if there's a reduction in the mortality rate of the phytoplankton there in the anoxic zone. So in this model, I have this SEM forming because there's lower phytoplankton mortality in the anoxic zone. How did I get that? So that's a further hypothesis, another degree removed from the mechanism. But the way that I was able to think about how to do that in the model was to have some oxygen limited grazing by zooplankton. So it's not saying that there's no grazing, it's just saying that perhaps there's slightly less grazing if some zooplankton predators are limited by oxygen. We know that from Karen Wishner's work and others that there is plenty of zooplankton grazing of bacteria and phytoplankton within the anoxic zone. But if there's just a little bit of shelter from predation, then there can be this maximum. So that's it. I went through this pretty quickly, but maybe that's good. So we've answered the questions: what allows for the coexistence of aerobic and anaerobic activity? Does it require vertical mixing? So we found that using this resource ratio theory in a simple system, that we can have stable coexistence of seemingly competing aerobic and anaerobic metabolisms, and that the ratio of those metabolisms is according to the oxygen to electron or supply ratios, not the concentration, but the supply. So further, what coexistence mean when you zoom into microbial cells? Is there variation in time or in space or both? So I've been working with a postdoc in Victoria Orphans group at Caltech to test out this in an actual chemostat, not just a virtual chemostat. And second, what forms the secondary chlorophyll maximum? So the hypothesis here that comes from a model that could be tested by observations is that there's lower mortality rates of phytoplankton at this SCM. And then the further hypothesis is perhaps it's that zooplankton grazers are limited by oxygen, but it could be other things, right? There could be less active viral lysis happening for whatever reason. So that's it. In the broader context here is that we've been able to use this redox chemistry to inform the parameterization of a very complex web of different microbial functional types. And so in this way, we've been able to simulate this anoxic zone biogeochemistry that has these recognizable profiles as being driven by chemical potential and physical transport in the environment. So this leads towards better predictive ability of our models ultimately. It also allows for emergent metabolic biogeography, the predictions of the amount of biomass that's happening, what biomass is responsible for which metabolisms in a way that we can ultimately connect to sequencing data sets. The fact that we don't rely on these oxygen thresholds that determine the presence or absence of any one metabolism allows for there to be this coexistence. So with that, I'll just thank the co-authors for all the projects that I mentioned, and shout out to Victoria and Yamini at Caltech and Andrew Babin, who's been ongoing conversation about denitrification, and a thanks to the Simons Foundation for supporting my postdoc work before at USC. And with that, I'll take any questions. Thank you very much.
Thanks, Emily, for the great talk. We have time for a couple of questions for Emily. Don.
Thanks, Emily. That was a really terrific talk. I have a quick question about the loss rates informing the secondary chlorophyll maximum. Could slower settling rates also contribute to that, being pro-chlorococcus?
Slower settling rates, like the rate of sinking of phytoplankton slows down in the anoxic zone?
Just their sequestration rate.
Yeah, I haven't thought about that. Yeah, so, right. So I thought about this like budget of phytoplankton biomass as being a function of nutrient supply and loss rate, but could there be no transport as well? Sure, maybe there could be. I think I have to think about that a little bit more carefully, but I'd love to talk to you more about that.
Okay, thanks. I think we have an online question now.
Yeah, hi, thanks. Emily, this may be a naive question from a physical oceanographer, but is there a role or a place for reactive oxygen species in all of this? Is there some way in which they play into this story, the superoxides? Thank you.
Yeah, right. So sure. So I guess the way that I put models together is I use the simplest version possible to answer the question that I'm trying to answer. So I think we could, I don't know a lot about reactive oxygen. Does it change the results of any of the things that I showed? I guess that's the question, right? I don't know. It's certainly a framework in which we could add, there's a variety of different ways in which we could make this model more complicated. And the key would be resolving it in a way that makes sense quantitatively. So we could, so the framework, right, can be expanded to any degree. So I guess that's all I can say about that, is that no, I haven't thought carefully about it. And I don't know that, but I have, given that we have producers key biochemistry, I don't think it would fundamentally change some of the answers qualitatively, but it sure could play a role quantitatively in predicting the exact numbers of things. So that's my sense. Thank you.
Okay, let's thank Emily again. So our next speaker is Peter Kroll from the NEOS.
Yeah, can you hear me okay? Yeah, okay, so thank you very much. No, we've been here for 9 hours, so we'll be here for a little bit longer. I do want to thank everybody for sticking around. Yep. So I want to thank the organizers for this opportunity to speak, and also for the previous, I think, 5 or 6 talks, which delved so deep into the nitrogen cycle that I don't have to say anything about that anymore. There is a bit in there though. So what I would like to do maybe is to step back a bit from really microbial processes and see how we can maybe link sediment and water column and link nitrogen and phosphorus a bit in a pretty interesting upwelling system, the Benguela upwelling system. I want to thank my co-authors, which are listed here. Without them, the data would not be there and the interpretations neither. And I want to emphasize that this work is really done by Kristen Ungerho PhD student who works on phosphorus biogeochemistry, and Zoe van Kemenade, both at NEOS, who works on nitrogen biogeochemistry. And this was from a cruise in 2019, where you're dependent on a lot of people to make everything happen, from sampling to processing everything. So that's the crew for our cruise, who are also acknowledged. So here is not the Brightberg plot, but what I thought I'd illustrate here a bit is we have satellite-derived chlorophyll A. Maybe I should use the laser pointer. Pointer laser. No. Yeah, there we go. So here we have chlorophyll A. So we see these productive upwelling regions. We've talked about this so much, I'm not going to spend time on this at all. And then another plot of oxygen minimum zones, where actually, if we dive into it a bit, I mean, we've talked about these oxygen minimum zones sort of as, look, it's purple there. This is an oxygen minimum zone. Now we're going to look at how bad the low oxygen is. The Benguela one, the area that we were in, is not as bad as these fully oxygen depleted zones that we've seen before. Around 50 micromole per liter of oxygen, but it's an interesting system that has quite low oxygen. So that's why we study that one. So here we have this nitrogen cycle, again, a new plot. It's been talked about so much. So I think the only thing we need to focus on here is the fact that if you go to this area, which is Boxic, Don Canfield, what we see is that these nitrogen loss processes start to occur, and you start to lose, which was nutrient nitrogen. It leaves the system as dying. I mean, we've had 2 hours explaining this, and I know very little about it. I just thought I'd also include a pretty picture where you see this. This is from a model which converges with data. So it's a really robust model where you see nitrogen loss rates from Anamox and denitrification, both in the water column and then in the sediment. Obviously, in the sediment, it's all over the place because oxygen does not penetrate very far into the sediment. And you see these hot spots, which are the coast, where you have a lot of organic matter in your system and oxygen penetrates even less efficiently. So for phosphorus, so if you look for oxygen-dependent nitrogen cycling, you beautiful plots. If you look for oxygen-dependent phosphorus cycling, it's pretty scarce, but I like these really old ones from the late 80s and also the 90s. From Gechter and others, which there are many details that we can argue about. But what it does illustrate nicely is you take an aquarium, you add some sediment, you add some water, you oxygenate the water, and you see that your phosphate is relatively low. And then you deoxygenate the water, you see this anoxic period, and comes out of the sediment together with iron. You re-oxygenate your water, which you see here, and it all goes down again. So this phosphorus itself is not directly dependent on redox processes, but because it's so strongly associated with oxic phases in the sediment, like iron oxide, it reacts very strongly. And then this was succinctly, I think, illustrated in this plot where you see you have anoxic sediment at depth. Like I said, at depth, you always have anoxic sediment in many places because oxygen just does not penetrate that far in the sediment. This phosphorus diffuses up and is then taken up by either bacteria or it's absorbed onto iron minerals. If your surface sediment is also anoxic, which happens if it's overlain by oxygen-depleted waters, these sequestration processes break down and this phosphorus is released again, which can then trigger eutrophication and deoxygenation. So this is, for instance, in the Baltic Sea, one of the big problems, this continuous recycling of phosphorus. It's not being buried, it's available all the time to fuel these blooms. So nitrogen and phosphorus are both key nutrients. They both respond very strongly to water column oxygenation and sediment chemistry. That's why. So if we look a bit at where we went, this is off the coast of Namibia in the Benguela upwelling system, where we had a transect of stations from the slope to the shelf and also a few along the shelf. And we tried to capture like a bottom water oxygen gradient. That's the idea. Like what happens to this coupled cycling, this exchange between the sediment and the water if you move from well oxygenated to poorly oxygenated systems. And you see that we capture that quite nicely with our stations. We have a few that are around zero, and then you move, as you go to deeper waters, you get more and more oxygen. If we look at this plot in like a transect, what you see is here we have the shelf and here dead. So this is kind of nice because what we see here is that we have our OMZ, so this oxygen minimum zone, which is fueled by upwelling of nutrients, which are then used for productivity. But we see it's not very strong. It's like there's still about 50 micromolar oxygen in that one. So the Benguela oxygen minimum zone in this area is not very impressive. But what you also see is the shelf is then fed by these already oxygen depleted waters from this zone, and there's high productivity, and your shelf goes anoxic. So we have kind of a nice system of a perennial oxygen minimum zone and then a shelf that is seasonally strongly anoxic. And just a bit about why it's not such a strong oxygen minimum zone is if we look here, so now we're looking basically, let's say along latitude, you see that from the south, there are these relatively oxygenated waters moving up. So our system is in the area where these oxygenated waters with the red colors are still quite dominant, and they basically supply enough oxygen not to go fully anoxic. But if we then focus on the shelf, so we had a station here, station 6, which was on the shelf. And if I would like to draw your attention to the oxygen profile here. So we have close to saturation, but it's not actually saturated. So these waters are so strongly oxygen depleted, and the oxygen depletion is so strong that even your surface waters are relatively oxygen depleted. And it goes down very strongly, and then below, sorry, there's no depth scale here. It's about 100 meters, but about below 30 meters, you have very little oxygen. And what you see there is that you have your nitrate, and that also disappears quite strongly. We've talked about the loss processes quite a bit so far. And what we see is also this nitrite peak here. So denitrification processes seem to be a role. And then I had colleagues, Zoe, for instance, she worked a bit on what the role of Anamox may be. And here we see that during this season that we were there, these intact polar lipids, biomarkers for active Anamox, they were not really present. There were remnants of Anamox bacteria. It happens there every now and again, just not then. I think this links back also to what we talked about earlier, that you can find these signals or you can find microbes because these conditions on the shelf are so dynamic. This is also like a time shot. So then what you can do, which is kind of nice, is you can calculate the nitrogen deficit, which many of you are probably familiar with. And the idea behind it is that organic material, algal organic material, has a carbon to nitrogen to phosphorus ratio of around 106 to 16 to 1 on a molar basis. And the idea is that both in organic material and in the surrounding water, this 16 to 1 ratio between nitrogen and phosphorus, you can find it in most places. There's variability, there's geographic variability, but it's rather robust. So what you can then do if you want to see, okay, how much nitrogen am I losing from your system? You say, well, I have this much phosphate, kind of assuming phosphate doesn't do that much, times 16 is the amount of nitrogen I should have. I subtract from it what I actually find, and the number I get is the deficit. And what you then see at these stations is nitrate goes down. There is a bit of ammonium, I'll show you later, but we see nitrogen deficit appearing. So it seems that we have nitrogen loss by microbes in this system. But I don't care that much about nitrogen. So if we then look at the profile, what we see is again the nitrate, which goes up and then goes down very strongly in these waters. We see in orange ammonium accumulating, but there is nitrogen loss. But what we also see when we talk about this nitrogen deficit, which I find kind of interesting, is there is a net increase in those bottom waters in phosphate. Now, I've already said that there's no real direct redox dependence of phosphorus. There are no, in the water column, we don't really know of bacteria that may be sequestered. You don't as a gas or anything. So if I see something like that, you would say, okay, maybe being supplied from the sediment, which is good for me because the sediment is where I do most of my work. So during the cruise, we also collected some of these sediment cores and tried to figure out, okay, so how are sedimentary phosphorus cycling processes working together with water column nitrogen cycling processes to shape these nutrient profiles that we find. How do we interpret these? And then we have a few pore water profiles. Again, I'm very sorry. I realized that earlier on we had these beautiful presentations with sharks, and there were movies and videos, and then there's a stage and you get pore water profiles. But I think one of the really strong sides about these pore water profiles is they tell you about active processes. So if you look at the minerals in your sediment, you see what ends up in the sediment. But if you look at the pore water, what is being dissolved, what is being precipitated, you get an understanding of what is happening right now. And then what we see is this DIC, which is in millimolar, actually, sorry, it's quite high. This says something about carbon degradation. And then as carbon degradation, DIC, you see ammonium being released as well, and you see phosphate being released, which makes sense as this organic matter breaks down. But what we see in the sediment, even more extreme than in the water column, is a very low ratio. So what seems to be going on is that there's not only just algal organometallic breaking down, supplying dissolved ammonium and dissolved phosphate, which could then escape from the sediment, which I will talk about later, but there seem to be contributing phases. And then here, I also have fish in my presentation, thoroughly dead. The idea is that fish bones in sediments that don't have a lot of oxygen are quite well preserved, and then they start to dissolve quite quickly in the sediment. And fish made up of calcium phosphate. So if these bones dissolve, a lot of phosphate is supplied, in addition to the stuff that comes from normal organic matter degradation. So we get really high phosphate, really low N/P ratios, telling us that the sediment is supplying even more phosphate than you would expect based on just organic matter. And what we also see is that right at, and I mean, these profiles are very freaky said about it, but what we see is immediately at the sediment water interface, should also be millimolar, sorry, we have thousands of micromoles per liter of sulfide, a lot of hydrogen oxygens being generated from sulfate reduction, as you see here. So it's an where this strongly reducing sediment just has all of these nutrients and toxicants just sitting there at the sediment water interface accumulating. And that can be, if we look at sediment and the water column, that can be quite interesting because these can all escape from the sediment, which I'll talk about later a bit. But here comes the role of microbes as well. So what we've seen is in 6, where we have this of ammonium, of phosphate, and a lot of sulfide. And then station 4, which is the same depth, pretty much same sediment type, lower than 5 micromoles per liter. But there we see that the sulfide accumulation is really suppressed, and it only occurs deeper down in the sediment. And what we saw in these cores was in the surface sediments, you saw all of these sort of white filaments for oxidizing bacteria. This role of microbes comes in within a sediment context here. And so these microbes are oxidizing the sulfide. So hydrogen sulfide is being transformed into elemental sulfur, which is not toxic and not mobile. And so basically it's putting a lid on this sediment and avoiding a release of toxicants. So we have done incubations to measure the rates with which these sediments are emitting all of these relatively important nutrients. We have some struggles with some of the data. So I'm presenting here the diffusive fluxes. We have some that really link nicely with the rates that we've measured, but I don't think that's for today. So what we see is at the bottom, you see the 2 sides. So the one with sulfur oxidizing bacteria, the whitish one, and the one without. And what we see is in the at the site where we have no sulfur oxidizing bacteria, you have a lot of phosphate leaving the sediment, so 1.5 millimole per square meter per day, a lot of ammonium and a lot of sulfide. And it's kind of cool because this links really well to different type of observation. And I think earlier someone also talked about these white clouds being observed on light image. I think it was also linked to this. So you see these sulfur plumes seasonally when the system is very oxygen depleted. You can see sulfur plumes in satellite images. So this is this pumping out a lot of hydrogen sulfide, which is not kept in the sediment by the microbes because they're not present in the site for some reason I cannot tell you. And then the sulfide is oxidized, elemental sulfur is white, and you see this here in the satellite images. So we have a very strong link between what happens in the water column and the sediment here, this benthic pelagic coupling. And then what is kind of nice here, I guess, is that it also links in maybe a bit to the dynamics of these systems, is that if you take a lot of these stations along the shelf, so these are all about 100 meters, all oxygen depleted, you can see that there's quite a bit of variability, and you have these spots. So this is just along where you see enrichments in phosphate in your deeper waters, and this is phosphorus being supplied to the water column from the sediment. And that translates it quite strongly to these strong nitrogen deficits. So my proposal here is that if you work with these nitrogen deficits as an indicator of the strength of nitrogen loss processes, think about phosphorus every now and again, and maybe it's supplied to those types of systems. Now then, let's take it a bit more boringly. Yep, I'm almost done. We go to a station which is here below the oxygen minimum zone. It's quite well oxygenated and everything becomes a bit more boring. Ammonium is not there present right at the sediment water interface, neither is phosphate, everything is quite low. So this site at the moment is keeping its nutrients in the sediment because there's oxygenated water overlying it. But we've also talked about the variability in OMZ intensity. So what we did here is take a 10-meter core and see, okay, if we go back in time, what can we see there? And I just have one slide about that. So, sorry, this is that long core that we take. And what we see is we take molybdenum because it's the best proxy for redox conditions. And what we actually see in this area over the last 20,000 years or something, there's been a strong decrease in molybdenum. So the system is much more oxygenated, oxic at the moment than it was a long time ago. And you see, so if we go back in time, it was more reducing, we have more organic carbon. But the interesting thing is the phosphorus coming from organic carbon actually sort of decreases, which fits really nicely with this idea that the recycling becomes more intense if the system has oxygen. And you see that the ratio between organic carbon and the phosphorus organic matter increases. But at the same time, so here's the link with the fish that we've talked about so far, is we see an increase in the deposition of these fish debris. And this is not just an interesting hobby. This is actually a very important sink for phosphorus in these sediments. So what we then see in these systems is you move to anoxic waters, a lot of this phosphorus from bacteria and iron minerals is being released back to the water column, but there's actually a new sink acting, making the phosphorus retention in these sediments more efficient, which then links into this whole idea of anoxic sediments supplying more phosphorus to maintain eutrophication and anoxia. So these are just a few conclusions, and they're mostly about the fact that these microbial processes affecting both nitrogen and phosphorus shape these water column profiles. And then nutrient stoichiometry, nutrient limitation will actually change if you change the, let's say, overall oxygen state of the oceans and if you intensify your oxygen minimum zones. And then we just have really complex feedbacks between the water column and the sediment. Thank you all for your attention.
I think we can take one very quick question for Peter. Yep.
Hi, thank you for your talk. So I was wondering, so when you showed that DIC and ammonia profile where it's increasing with depth and then decreasing, so that is beneath like oxygen depleted water, right? So like I was wondering like why there is a, the decreasing trend of the DIC and ammonia, because I wonder that bioirrigation won't be a reason for that.
The decreasing trend again at depth.
Yeah, at the depth.
Yeah, so this is where indeed you catch me, because the station is a bit weird. Like, these are not the profiles you're supposed to see if this is a steady state system where you just indeed have more reducing conditions and accumulation of all of these products at depth.
Exactly,
you're absolutely right. So the surface layer, there is something happening in the surface layer. And we see, for instance, a nitrate peak underneath the sulfide. So there seems to be some kind of pumping. So microbes are shaping all of these profiles.
For the DIC as well?
For the DIC, I don't know, but if you look at also the ammonium and the phosphate, there is just very little, let's say, being accumulating in the pore water there. So this top layer is different from what's below it, is the short answer. So yeah, that's a very sharp observation. Yep.
Okay, sure. Thank you. I might have some questions that I asked.
Yeah, just come and find me. Yeah, thanks. Thanks again.
Thanks again, Peter. So our next speaker is Geetha Plakal, who I think is joining us from the Alliance for Planetary Health, which might be located in Boston, I think.
Thank you so much. Can you hear me?
Yep.
Yeah, okay. And good evening to all. And today I present my topic on Land and Soil Remediation Project 2022. And background of the project. Sorry for delay. Extensive agricultural practices and use of artificial fertilizers that increasing day by day, and the demand for farming is rising with increasing population. But the use of artificial fertilizers leads to the loss of soil quality and nutrients. And this will lead to big economic loss and a reduction of agricultural production. Besides this, the leaching of excess nitrogen from farmlands will reach into the coastal and marine waters. This leads to increasing nutrients in the aquatic ecosystem and eutrophication and poisonous algal blooms. This will affect the water quality and triggering the marine dead zone formation in seas inverse by anthropogenic impacts rather than natural ways. And the dead zones are the areas of low dissolved oxygen content, and that is dangerous or fatal to organisms that wandering in that area in aquatic ecosystems or coastal zones or marine waters. And it indicating that we need to reduce the use of nitrogen based products and a sustainable way of farming techniques for a healthy environment. And we can check on evidence for this marine dead zone formation. Sorry, I am from a remote area, so that it will create some issues for me. Yeah, Gulf of Mexico hypoxia report from 2021 shelf-wide 35th hypoxia research cruise by Louisiana State University. And their major results, according to Professor Nancy Rabelais, research led at LSU Department of Oceanography and Coastal Sciences, the dead zone is 6,334 square miles or 16,400 square kilometers from July 25 to July 31 in 2021 year. This is larger than the findings of researchers based on river discharge and nutrient loads in May 2021. And this is the higher than average river discharges from the Mississippi River and the Atchafalaya River in July provided more fresh water. And this contributed to more algae, more sinking carbon, more bacterial respiration, reducing the dissolved oxygen to this region. And according to the 16th large hypoxic or low oxygen zone recorded by the researchers in the northern Gulf of Mexico, west of the Mississippi River in the 35-year-old record. And we can see based on their data here, it is the average for the year from 2015 to 2021 is greater than the earlier years in their research study. and their recommendation. The nitrogen recommendation to avoid this issue. The nitrogen loading of the Mississippi River to offshore remains high, and the researchers recommended that the use of nitrogen-based products need to lower to overcome this issue. And what we can do to mitigation activities against such marine dead zone area formations in ocean, and it need to be based on the principle of health, ecology, fairness, and care. And the objectives of our research or our study, the purpose is to enhance education, health, ecology, economic, and social activity through learning, and to provide a way forward in soil remediation and a sustainable farming and economic and ecological development, and to prevent excess consumption of toxic substances like insecticide, algicide, and herbicides, and to provide high quality food in a regional or global market, to remove the harmful artificial fertilizers, pollutants, and to mitigate marine dead zone formation, eutrophication, and poisonous algal blooms in approximately 464 spots identified worldwide in an ocean or marine ecosystem. And to mitigate the ocean acidification and climate change induced impacts on marine life by interaction of marine dead zones, and to conserve marine biodiversity and soil biodiversity. The introduction. Our research is to support economic development of communities around the world through sustainable farming for a better future. It is a humic acid agent in coal ash-based material to create a time-released product in soil to grow microorganisms to symbiotic relationship with plant roots named vital grow. It allows nature to work on root system of the plant rhizomes, trees, or other biomass to assist a symbiotic relationship is formed through human interactions. This product is non-polluting or twofold CO2 uptake through natural sequestration and healthy soil environment. Microbial fungi to meet soil nutrient requirement for the plants. No salts, no leaching or alteration of DNA in soils. By increasing the ratio of potassium to sodium in soil solution, plant tolerance to salinity increases. Our purpose is to establish vital grow, that is the product name of our company, and as a benchmark methodology to protect soils and water interconnection processes, and to include seas, gulf, and oceans and their conservation. Methodology. Our methodology is less expensive and sustainable way of soil remediation method with proven records. Methodology by Ali Khalwati 2010. It is suitable for different types of soils, and a reverse NPK artificial fertilizer urea by using symbiotic relationship method, and positive combination of potassium and zinc against salinity. Coal ash is used to obtain humic acid, which is derived from the organic carbon in coal. The rate at which humic acid is obtained from coal has led to a great deal of attention being paid to the organic matter derived from coal in sustainable agriculture. We use the waste stream of burned coal, which benefit to humanity and the Earth's or planetary health. Humic acid can be used to reduce the use of chemical fertilizers because the connection between humic acid and nitrogen phosphorus prevents over-leaching of nitrogen and phosphorus. Organic fertilizers containing humic acid as a disinfectant and plant hormones in plants. Biofertilizers reduce the nutrients needed for the pests and eliminate them on the farm. It recommended an integration management of sustainable agriculture method with the use of biological and organic fertilizers will reduce the mass of plant pests and on the other hand bring healthy plants to the production stage. Function and role of potash in plant. Potash contributes to the quality of the fruits and the formation of protein in the fruit and reduces diseases in the plant. Potash helps ornamental plants to flower and fruit trees to enlarge and make the fruit colorful. The use of microorganisms or the significance of microorganisms in such biofertilizers. Bacteria prevalent soils also have high nutrient availability and enhanced nutrient levels as a result of more rapid organic matter decomposition. They are the primary decomposers and dissolving phosphorus and making more available to plants. Abundant in the area immediately next to plant roots, called the rhizosphere, and where cloud of cells and chemicals released by roots provided ready food sources or leaves, photosynthesis and sugar production. Provide nitrogen through fixation, detoxify harmful chemicals or toxins, suppress diseases of organisms, and produce products that stimulate plant growth. Source of most of the antibiotic medicines we use to fight diseases from such microbes. And the mycorrhizal fungus increases nutrient absorption in plants from 10 to 100 times and uptake of nutrients increasing due to increasing porosity in soil for plant root gain. Azotobacter helps to absorb inaccessible substances to plants and allows the use of nitrogen, phosphorus, potassium to take place naturally by plants. And the bacteria and microorganisms in inorganic fertilizers act like antibiotics and cure plant diseases. We can see in this picture the action of such fertilizer in plants with the help of the atmospheric nitrogen fixation by root nodules, bacteria, and the abscessular mycorrhizal fungi association and other soil microorganisms and etc. And major advantage the product: complete correction of biological life of soil after 2 years of after using, and increase plant resistance against frostbite, dehydration and drought, and reinforcement against pests in order to make nitrogen in plant, and increasing amount of soil materials and help plants absorb them, and helps to absorb non-absorbable soluble material in trees and plants, and accelerate NPK absorption and improve soil's biological and health conditions, increase growth and production of 30 to 40% in garden plants, crops, ornamental greenhouse flowers, and it increases the amount of iron, manganese, phosphorus, potassium and calcium, which are mostly non-absorbable in the soil for the plant. It stimulates growth of root, stem and branch strength, improves flower formation, seed production, improves the nutritional quality of the plant, and increases plant production by 30%. And the benefits of this organic fertilizer, and let's check this in a short way. In a soil desalination and water absorption and storage, and a reduction of acidity absorption of soil, and reducing water consumption in agriculture, prevention of pests and diseases, contains natural microorganisms and acetobacter functions in soils, and increasing rhizogenesis 8 times more than normal plants, and aeration, and it will increase along with porosity and permeability, and contains about 60% of organic matter that is completely soluble in water, increasing soil moisture holding capacity, becomes spongy, and that is another benefit is the water conservation by reduce the use of irrigation for farming.
Sorry, Geetha, we're close to out of time. Can I ask you to move towards your conclusion, please?
Okay. Optical amount of in agricultural soil vital growth fertilizers analysis results is this is the that contains nutrients combinations or concentrations, and 2 or 3 slides only, and this is a list of that nutrients and its concentration, and the fertilizer application rate in flowers and ornamental plants, and it's in a ornamental plants with bulbous 500 kilogram per hectare, and ornamental plants with spring roots 1000 kilogram per hectare, and it is in the flowers and ornamental plants with tuberous roots with 800 kilogram, and all these fertilizers advantage is promotes the longevity of ornamental flowers, increases the resistance of flowers to heat, prevents pests from attacking plants, and another one is fertilizer application rate in 2-year-old seedlings per this is the details of that portions, and
okay, Geetha, I'm going to have to talk now.
Okay. It's just concluded. I'll conclude.
Okay, thank Thank you.
Yeah.
I'm afraid we don't have time for questions, but thanks again for your talk.
Yeah, thank you so much. Thank you.
And thanks again to all the speakers.